Hello, champions, and welcome back to PW English channel. So, here we are going to discuss a very important chapter of zoology, and that is biomolecules. I am your Diksha Sharma, ma'am, and today I am going to teach you all about biochemistry. Now, what is a biochemistry? So, you must have heard of this word biochemistry. This is actually a subject which compiles of both biology and chemistry part, right? So, all the chemistry that is associated with the living organism, that is biochemistry. If you are going for the higher studies in the medical field, like you're doing MBBS or you're doing BSc or BS, anything like that, you will come across a chapter, or a subject. Basically, you will come up across the subject in your higher degrees, that is biochemistry, and there you'll be learning all these things in detail. So here we are going to talk about the biomolecules. What are they? What are they about? So trust me, it's very important and very interesting. You don't need to be scared about this chapter. A lot of students, they're so much scared about this chapter. I really don't understand why. Because now, if you follow this chapter with me accordingly and appropriately, this is really going to be very easy. So let's get started. The biomolecules. All right. So first of all, what is a biomolecule? What is a biomolecule? Bio means living, and molecule means molecule. The one you study in chemistry, right? So biomolecules, it's all the molecules present in living organism. Present in living organism. So in the NCRT, you will come across a definition in the another form. What this says, all the organic compounds present in living organism are biomolecules. That's also true, but you just cannot forget about the inorganic one as well, right? So organic are the elements which are made up of carbon, whereas inorganic are the one which are like sodium, potassium. They're also present in our body, so you cannot ignore them. So this includes both organic, all right? This includes both organic as well as inorganic elements. Fine. And the study of these biomolecule is biochemistry. Is biochemistry. All right. Moving further to how to analyze the chemical composition. For example, if I say I have some tissues, right? I have a plant tissue, I have animal tissue. So how am I going to know what is present in that tissue? You must have done some experiment, or some scientist must have done the experiment. That's why today you know that we are made up of carbohydrates, proteins, amino acids, and everything like that. So there is a very simple experiment that is that's a primitive experiment that actually tells you what is present in your living tissue. So how this experiment is done, let's get started. So first of all, we take a living tissue, and this living tissue can be your plant tissue, it can be your plant, or it can be your liver, animal's liver, right? Then what you do is you crush and grind them, or grind it in mortar and pestle. In mortar and pestle, in trichloroacetic acid. In trichloroacetic acid. So what we do next is we take the tissue. We actually dry the tissue first, we take it, and then we'll crush it in the mortar and pestle. You must have seen the mortar and pestle. That's also present in our, Indian homes to crush the ginger or another masalas or spices. It's just like this, right? And it has a little instrument with it so that you can just put anything here and then you can crush it. But here we are also adding one chemical. And what is that chemical? Trichloroacetic acid. Do not forget about it. So what we are doing, we are adding the trichloroacetic acid. We are grinding it. We are grinding this chemical. Now then what we are doing? After we have, we have grinded it so nicely, then we will filter it. Then what will we do? We will filter it. Now, how are we going to filter it? You must have seen a lot of, all these equipments in your laboratories of school. Or if you have not seen it, I'll just draw it and then you'll come to know. You also draw that in your practical notebooks. What's that? That's a beaker and then it's a funnel, right? So what we do is we take a beaker, we take a funnel, and we cover this funnel with a cheesecloth or a filter paper or a cotton, right? So what is this? This is a cheesecloth, the muslin cloth or cotton. So we are using this cheesecloth or cotton as a filter, right? So when we put this whatever we have made, right? This is in the form of a slurry. Slurry means that is having, some little viscosity. So what we have done is when we have grind this one, now we are going to filter it. And what is it? It is slurry. Slurry is all that is formed after grinding, that viscous solution type. So we added slurry here. After we have added the slurry, it is going to filter. Some of the things which are not going to filter, they will remain here. They will remain here as retentate. Now, what you will call it as retentate. Not everything get filtered. Fine. And the thing that get filtered easily here, that is the filtrate. What is it? It's the filtrate. So after filtration of the slurry, you will get two components, the one which is present in the beaker, that is came down with the acid, right? And the one which has just left behind there in the filter. Like whenever you make laminate at home, you filter the laminate with that sieve. In the sieve, some of the lemon seeds are left. So that lemon seeds that are present in the sieve, that is your retentate, and the solution that's in your glass, that the laminate, that is your filtrate. Fine. So now what happen is, what is this retentate and what is this filtrate? Or what are the components that are present in the filtrate and what are the components that are present in the retentate? All those components which are larger in size, they are not able to get dissolved in this acid. The one which are insoluble in the acid, they came in retentate. Why they were not soluble in the acid? Reason number one, they might be in the larger size. They might be of the larger size. That's why they're not able to, get dissolved here. Second, they must not be like molecules. For example, if I talk about lipids, the lipids, they are no friends with trichloroacetic acid. It's not soluble in trichloroacetic acid. If it is not soluble in trichloroacetic acid, then it will remain in the retentate. Whatsoever is there in the filtrate, it is soluble in the acid. So the one which is in the filtrate, we say it is the acid soluble fraction. It is the acid soluble fraction because it was able to solubilize in the filtrate in the acid. That's why it has came down with the acid. But the retentate, this consists of acid insoluble pool or the insoluble fraction. So here all the macromolecules and in micromolecules lipids, they are acid insoluble and they are the part of retentate, whereas the micromolecules are in the filtrate. So lipids are an exception here. That's why we say lipids are not strict macromolecule. There is a sentence which says lipids are not strict macromolecule. Why? Because according to this experiment, a lipid was found. The lipid was found in the retentate. When we performed this experiment, we saw that lipid is a part of a retentate. Lipid is a part of retentate. And according to this experiment, all those things which are a part of retentate, they are what? They are macromolecules. We suppose that if it is in the retentate, it must be a macromolecule because it's larger in size. That's why it is not the part of filtrate. But when we saw lipid there, it was like we were surprised why lipid is there, because if we see according to size, the lipid is a micromolecule. If we see according to size, the lipid is a micromolecule. But if we see according to this experiment, the lipid falls into the retentate fraction or the acid insoluble pool, and according to it, it should be a macromolecule. So why it's a part of retentate? Because lipid, whenever you crush the tissue, the lipid is present in a cell membrane, right? So how is your cell membrane? It's the double layer, phospholipid double layer, right? So when you crush it, all these fatty acid, they form these small micelle-like molecules. These micelle-like molecules. When you crush the membrane, that membrane forms some micelle-like molecules. So it's a micelle of lipid. And this is acid insoluble. This is acid insoluble. So that's why lipid, we say it's not the strict macromolecule. Why? Because according to molecular weight, lipid is a micromolecule. But if you see according to this experiment, it falls in the category of retentate. Why? It's not because of size. It's because it is insoluble in the acid, because whenever you crush the lipid, it will form this micelle-like structure, which is acid insoluble. Fine. So I have told you there is a criteria by which we say some are micro and other are macromolecule. All right. So all the molecules which have molecular weight, molecular weight of around 1000 Dalton, around equal to or less than 1000 Dalton, it's a micromolecule. So all your simple sugars like glucose, like your amino acids, right? All these according to molecular weight, they are micromolecule. What are they? Micromolecule. Even lipids, because lipids molecular weight never exceed, 800 Dalton. It's never more than 800 Dalton. So according to molecular weight, it's a micromolecule. But according to that it's a macromolecule. So that's why we say the lipid is not a strict macromolecule. Otherwise, your nucleotides. What are nucleotides? These are the building blocks of your DNA. We'll be studying them in detail in this chapter. Just look at the only words now. And what are biomacromolecules? These are molecules with molecular weight of more than 10,000 Dalton. Right? So if anything is having the molecular weight of 10,000 Dalton or more, it will be biomacromolecule. Alright, so now what is this DA? DA is Dalton. What is Dalton? For example, whenever we weigh something, you weigh it in the kgs. Just like that here in the molecules, biomolecule, we use this Dalton, right? So here, what are the things that come under the category of the macromolecule? These are polysaccharides or complex sugars. Then we have proteins. Then we have the nucleic acid. What are nucleic acid? Your DNA, RNA. So I'm writing example here, DNA. So all these fall under the category of macromolecule, and these fall under the category of micromolecule. All right. Usually, the monomers, they are micro, whereas polymers, they are macro. For example, protein is made up of building blocks that are amino acid. So amino acid is a monomer, and what's a polymer? Polymer is protein. So whenever something is polymer, it has a lot of units. That means obviously its molecular weight is going to increase. That's why it comes under the category of the macromolecule. Next we have ash analysis. What is ash? Whenever you burn someone after the death of a person, which we say cremation. You see, after cremation, only some part of the body is left. Otherwise, everything gets vanished there, right? So what happen is, whenever you burn a tissue, you completely burn a tissue. Completely. I'm talking about completely. The organic part, which is a carbon containing, it will evaporate in the form of CO2 and water. Whatever left behind is the ash, and that ash contains all the inorganic things like calcium, like sodium, and all other components, right? So, here, what is ash? We took a tissue, we took a wet tissue, and then we wait, okay? Then we wait. So, we took the wet weight, and then what we does, we actually keep it in the oven. We want to dry it, right? And then we dry the tissue, like we put it in the oven, or we just kept it under the sun, so that every extra water gets evaporated. So then we again wait, and that's the dry weight. So here we took the wet weight and the dry weight. So after we have evaporated the excess water, now we are going to completely burn it, completely burn the tissue. So when we completely burn the tissue, now what happened here is, here happens two things. One, the organic, whatever organic is present, organic compound will evaporate in the form of CO2 and water, right? And what is left? Left behind is ash. Now, what does ash have? It contains inorganic compounds, right? So whatever ash have is the inorganic compounds. Fine, I hope that's clear to you. So after we completely burn the tissue, we analyze what this ash have. Ash is that black in color, right? So when we analyze the ash, we come across a lot of inorganic components or elements in the ash. So that means we are not just made up of the organic compounds. We have a lot of inorganic compounds as well. All right. Moving further to the next. So what does our body have? And here is a comparison with the Earth's crust. So this one is of the Earth's crust and that one is of the human body. So first of all, hydrogen. The Earth crust have 0.14 and human body have 0.5. Carbon 0.03, 18.5. Oxygen 46.6, 65. So you can see the most abundant one here is the oxygen. So what type of a question can arise from this chart? Either a direct question of what percentage of this component is present in the human body, or the most probable question is, which is the most abundant element in the human body. Then what will you say? You will directly say it's the oxygen. Fine. In fact, in the earth crust as well. Or the second abundant one. So if you talk about the second abundant one, that's carbon. Then nitrogen, it's very little in the earth crust. That's why nitrogen fixation takes place with the help of symbiotic association in the plants. Then we have 3.3% in the human body. Sulfur 0.03, here in human 0.3. Sodium 2.8, 0.2. Calcium 3.6, 1.5. Magnesium 2.1, 0.1. And silicon 27.7, negligible. So we don't have silica, right? We don't have silica. But silica to us is somewhere is also harmful. Silica in different ways it can be harmful. One of its product, can even cause cancer. It can also cause a cancer of lung or the cancer of skin. Fine. All right. Moving further to the next. Let's move further. Now, when we analyze the ash, so these were the various inorganic components that came out. Like we have sodium in us, we have potassium, calcium, magnesium, water, and other compounds. Now, so what does the sodium and potassium has a role in our body? First of all, sodium helps in osmotic balance. It helps in osmotic balance. Fine. Sodium and potassium both, they plays a very important role in nerve impulse. Next, calcium. Calcium helps in blood clotting. It also helps in muscle contraction. Talking about magnesium, magnesium act as a catalyst. It act as a cofactor for various enzymatic reactions. We'll study the cofactors in later section. These are certain elements without which the enzyme is incomplete, right? So magnesium plays a vital role here. Also, magnesium act as a catalyst for various chemical reactions. Water is the medium of all the chemical reactions. So you need something where you can dissolve all the things and you can make it. Like for laminate, you need water to mix up everything. Fine. So what is the medium of all the chemical reactions in a body? That's water. So it's the medium of all chemical reactions. Then we have various other compounds like NaCl. NaCl, it helps in osmoregulation. Calcium carbonate, in fact, we have calcium phosphate. That's the important part of your bone. Then we have dibasic and monobasic ions. These you can see here. What do they do? The dibasic or monobasic ions in blood, they regulate the pH. And the things that regulate the pH, they are known as buffers. So they act as buffers, right? So what are buffers? Which regulates pH. Which regulates pH. I hope that's pretty clear to everyone. So these are all the functions of various inorganic elements in your body. Actually, these are very important functions. So these are also biomolecules. Next, what are primary and secondary metabolite? Now, what are these terms? So, whenever we are talking about metabolism, there is a word known as metabolism. I hope a lot of you must have come across this word in your biology. So, first of all, let's talk about basics. What is a metabolism? So, whatever chemical reaction is occurring in your body, its all total is metabolism. So, it's the total of all the chemical reactions in body. Right? For example, you are eating food, the food is getting digested, or in your cells, the glycolysis is occurring. So, all these total sum of the chemical reaction, that is your metabolism. Now, all the components that participate in this metabolism are metabolites. For example, glucose is a metabolite. Glucose gets converted into glucose 6 phosphate. That's also a metabolite. Fine. So, all the molecules that participate in metabolism. What are they? They are known as metabolites. What do you call it as? You call it as metabolites. Now, metabolites are of two types. Some of them, they will be very important. Without them, the body is incomplete. And another, which are not that much important. So, whenever I'm talking about the primary and secondary metabolites, straightforwardly, I'm talking about plants and bacteria. In our case, in human body, there are no such thing like primary and secondary metabolite. For us, everything is important. So we have everything as a primary metabolite. Fine. But when I'm talking about plants, there are certain things which are very important, like glucose, right? Like all the enzymes in the photosynthesis or the chlorophyll. These all are very much important. Without them, the plant can even die. But if I talk about other things like your alkaloids in plants, they are secondary metabolite. So primary, which are very much important. Without this, a plant can die. Like chlorophyll, right? Or components of various cycles, whatever cycle, like C4 cycle, C3 cycle, whatever cycle. And secondary, these are not much important. These are usually the byproduct of primary metabolites. What are they? They are byproduct. What are byproduct? It's like it's formed during the chemical reaction, but it's of not importance. Now, what are the various secondary metabolite? First of all, whatever example I'm going to tell you of secondary metabolites, you actually have to learn it. You actually have to learn it, so with so much of of your interest, and you have to just keep it in your memory for a longer time. Why? Because in NEET, that's one of our favorite topic from biomolecules, so you have to learn it. Okay, so first is pigments. So there are a lot of pigments present in a plant. Like one pigment is chlorophyll. Now, other pigments are also there. You must have done it in the botany. Carotenoids are present in the carrot. Anthocyanins. All these are not performing the main function, that's photosynthesis. But yes, they are performing little function. But if we take out these pigment from a plant, the plant may survive, right? So that's why it's a secondary metabolite. Alkaloids, we have morphine and codeine. Morphine and codeine for plant is useless, but for us, it's very important. So whatever secondary metabolites have been known so far, we are using them for our purposes, right? So these are used for human welfare. So these secondary metabolites, they are used for human welfare. Fine. Okay, moving further to next example. Morphine and codeine, where do we use it? Morphine is a powerful, painkiller, and codeine is used in cough syrups. Terpenoids, monoterpenes, and diterpenes. Essential oils like lemongrass oil. So you must have heard of the nowadays the essential oils like tea tree oil. A lot of girls used to use it on the skin, and they're so much good for acne. That's of anti-acne properties. So likewise, a lot of essential oils and terpenoids have been discovered, which actually also protects the plant from the other insect or something like that. For example, if I talk about the lemon secrete a type of a terpenoid in a form of aromatic compounds that it will not allow other insect to come there and infect the plant. So terpenoids are like those only. Toxins like abrin and ricin, these are the toxins secreted by the plant so that other animals should not eat them. For example, if there is a cow coming down there to a grass, and grass wants to have some defense mechanism, so that grass is secreting that toxin, and whenever the cow eat that grass with this toxin, the cow will die. So likewise, animals have also learnt in the evolution that this plant they want to eat and this they should not be eating. And the plants, likewise in evolution, they have also produced these self-defense techniques in them, right? Or they have evolved with these self-defense techniques. Next, lectins, concanvalin A. This is in bacteria. This is completely useless to a bacteria. It's just a glycoprotein on the wall of bacteria. If this is a bacteria, it's just present like this in bacteria. To a bacteria, it's completely useless. But our body's immune system is so much clever. So whenever the bacteria comes inside our body's immune system recognizes concanvalin A, and hooray, the immunity or immune system starts working. So for bacteria, it's completely useless. Rather than protecting the bacteria, it is working with our this defense system, and it is letting a defense system know that this is bacteria. Then we have drugs, vinblastine and curcumin. Vinblastine is the anti-cancerous drug, whereas curcumin is present in the curcuma longa, which is turmeric, and it's a very good anti-inflammatory protein. That's why you must have heard, if you get some if you met across some accidents or something like that, or some injuries, your mom or your grandmothers used to say, just have turmeric milk. And that's so good because it has anti-inflammatory property. Whatsoever inflammation has occurred in your body due to injury, it will just vanish it. Then what we have, then the last one, we have polymeric substances like rubber, gums, and cellulose. For plant, these are also useless. That means plant can survive without them. So the thing is, yes, they're performing certain role in the plants. But if we compare them with the other metabolites, if you remove them from the plant can even survive without them. But yes, they are enhancing the plant's beauty, they are enhancing the other functions, but likewise they are not that important part of a plant's cellular metabolism. Fine. So these were secondary metabolites and they are very much important. To put a star, you have to actually learn them all. The previous to previous year question was this: which of the following is a lectin? So the answer was concanvaline A. So a straightforward question. So these type of question which are learning based, it's a hip hip hooray for you, because you can just directly see and mark the answer. For that you just need to learn it. That's it. All right, next we have carbohydrates. So now we'll talk about the carbohydrates. So that's the first organic compound or a biomolecule that we are going to talk about here. So let's talk about it. Carbohydrate. What does it mean? Carbo means carbon. Hydrate means water, right? So we say these are the hydrates of carbon. Hydrate means water. And why have we used the word water? Because the ratio of hydrogen to oxygen in water is 2:1. Same way in the carbohydrate, the ratio of hydrogen to oxygen is 2:1. So the ratio of hydrogen to oxygen is same in water and carbohydrate and carbo, right? Next, where does or where do they form? They are only produced naturally by the plants by photosynthesis. So they are produced, so they are produced by the plants by photosynthesis. All right. They are also known as polyhydroxy, aldoses, or ketoses. Now, why they are known as polyhydroxyaldoses or ketoses? Because they have poly. Poly means many. Many OH groups, hydroxyl groups, or some of them may be containing aldehyde group and some of them may be containing the ketonic group, right? But both these groups will never be together, right? In a normal carbohydrate, the carbohydrate can be a polyhydroxyaldose. That means it will be containing a lot of OH group and aldehyde, or it can be a lot of OH group with a ketone group. All right. So let's just see or let's just talk about them in more detail. So if I talk about the first one, that's glucose. Glucose formula is C6H12. All right. Okay. Before discussing glucose, let's talk about what type of carbohydrates are present. Okay, because that will make more sense to you to understand the later section. Carbohydrates are of three types. One are monosaccharides. Second is oligosaccharide. And third is polysaccharide. Now, what is the common thing in all of these? Saccharide. Saccharide means sugar. Saccharide means sugar. So in monosaccharide, you will be having only one sugar. In oligosaccharide, you will be having 2 to 9 sugar. And in polysaccharide, you will be having many sugars. So if I'm talking about glucose, glucose is one sugar. It's a monosaccharide. What is it? It's a monosaccharide. This monosaccharide is having how many carbon? 6 carbons, right? This is one sugar, one S. When these two joins, if the two molecule of glucose they join, that will become your oligosaccharide, disaccharide. With because it has two sugar, it will be known as the disaccharide. If I have a long chain of 1,000 these C6H12O5, right? I have thousands of them, then it will be a polysaccharide. Fine. So monosaccharide, since it's a monosaccharide, it's a single sugar. As I've told you, it has 6 carbons. Let's draw the glucose step by step. Follow me. Write 6 carbons. 1, 2, 3, 4, 5, 6. Put single bonds and mark the number of carbon. Like I say, 1, 2, 3, 4, 5, and 6. Draw this first, then we will move further. Draw this first, then we will move further. If I say the glucose is polyhydroxyaldose, that means it will be having the aldehyde group. The first carbon is known as the carbonyl carbon. Which carbon? Carbonyl carbon. Why? Because this is the one which is a main carbon and it forms the aldehyde group. What is aldehyde group? CHO. What is aldehyde group? CHO. So first carbon is the one which is containing functional group. What is a functional group? Due to which any compound is having its or it can perform its function. And that main group is the aldehyde here because it's polyhydroxyaldose. So that's why first carbon is a carbonyl carbon. So here, now you have to complete this structure. In the last glucose, write CH2OH because carbon has 4 valency, so you have to satisfy it. So you have 2 hydrogen, 1 OH here. Fine. On the third carbon, put H on the right side. On the third carbon, put H on the right side. On other carbons, put OH on the right side. Now, wherever there is OH, in front of it, put the H and satisfy the 4 valencies. Wherever there is H, put the OH and here H, right? So it's a game of HOH. In the third, its opposite, others are the same. And this is the structure of your glucose. Alright. So this glucose is also present in a ring structure. It's also present in a ring structure. Its ring is of 6 sides like a hexagonal ring. So let's draw the ring. So whenever a ring is formed, the first and the fifth carbon they joins. For example, I have this straight chain of glucose. Here is the fifth carbon. It is going to join with this first one and they're going to form a circle. As a result, if these two ends are joining, this will be out of the ring. This will be out of the ring. If these two are joining, this will be out of the ring. So let's see how the structure it looks like. All right. So if you can see here, this is a ring structure of your glucose, right? It's the ring structure of your glucose. All right. Just write H here. Okay. This is the ring structure. All right. So this is a ring structure of the glucose. It's quite simple. If I say this is the first carbon, this is the second, this is the third, this is the fourth, and this is the fifth, and out of the ring is the sixth one. In sixth one, you can see OH is on the upper side, so H will go down. H up, H down, H up. On the first one, there is a different criteria. On the first one, it's a different criteria. Why? Because first is a carbonyl carbon. First is a carbonyl carbon. If on the carbonyl carbon, only on the carbonyl carbon, OH is on the upper side of the ring. Listen to me very carefully. If on the carbonyl carbon, if OH is above the ring, this sugar will be beta glucose. What is beta glucose? OH is above the ring. Above the. And OH on which carbon? Of carbon 1 only. If I say OH is below the ring, then it will be the alpha glucose. So what will be the alpha glucose? OH below the ring. And what is the beta glucose? OH above the ring. But which carbon, which carbon we specifying here? Only carbon 1, because that's the carbonyl carbon. So these two structures, they are known as anomers. So what we say anomers? Anomers are alpha and beta anomers. Anomers are certain, compounds which are only different in one placement of one OH. You know how they are different? They're different only because of the OH. So that's your anomers. So we have two type of anomer, alpha and beta forms. In alpha form, the OH is below the ring, and in beta, it's above the ring. So this six-sided ring is known as a pyranose ring. Which ring? Pyranose ring. So whenever you come across this type of a ring in any biomolecule structure, you will say the glucose, it has a straight chain structure and it shows pyranose ring structure. What is a pyranose ring? Six-sided ring. Which ring? Six-sided ring. I hope that's pretty clear to everyone. Just come look across everything clearly. Hurry up. You just don't need to understand how the ring is formed. You just learn the structure carefully. That's it. Because when you try to learn it, you have to understand the electrophilic substitution reaction and a lot of things which is of chemistry. But trust me, learn the biomolecules according to biology. Because in biology, we usually learn things. When you tend to understand chemistry behind this, you will actually get confused, and then you will end up saying biomolecule is the most difficult chapter. Actually, it is not. The most interesting and the easiest. All right, that's the glucose. Similar thing which we are going to talk about, ribose. Like glucose was a 6 carbon sugar. This one is a 5 carbon sugar. Ribose is a 5 carbon sugar. So let's draw the ribose. All right. So ribose is a 5 carbon sugar. All right. So what's the common formula? Like I have told you, it's C6H12O6. So there must be a common formula for all the carbohydrates. Yes, there is. You use CN, H2N, ON. If I have 6 carbon, it will be C6H12O6. It is a 5 carbon, then it will be C5H10O5, right? So it's a common formula for all the carbohydrates. So if you know the number of carbon, you can reduce the formula. Now what it will be? C5H10O5. Let's draw it. Similar manner, draw 5 carbon in a chain. 1, 2, 3, 4, 5. Label them up. 1, 2, 3, 4, 5. If I say it's also polyhydroxy aldose, it's also the aldose, then what you have to do, you have to draw the aldehyde group on the first carbon, and first carbon is a carbonyl carbon. Then on the last put CH2OH. It's quite simpler because here all the OH are on the right side. Alright, so that's your ribose. Like your glucose, it shows six-sided ring. The ribose, it shows five-sided ring, and that ring is furanose. F for 5, F for furanose. So what is a furanose ring? It's a five-sided ring. Let's draw. Why ribose is so much important to us? You will get to know. So here, since it is forming the bond between first and fourth, like in glucose, it was first and 5 and 6 was out of the ring. Here, the fifth one is out of the ring. All right. So fifth one will be out of the ring here. So here is H. Here is H, OH, H, OH. And then we have to decide. If I say this is the first carbonyl carbon, second, third, fourth, and fifth. Now, if I want to make it as a beta ribose, what I'll be using? I'll be doing OH above the ring, H below the ring. So that will become a beta ribose. Ribose sugar is a very much important sugar because this is a part of your RNA and a lot of nucleotides in your body. It's also the part of ATP. Adenosine triphosphate is actually a nucleotide. And there the sugar used is ribose. Whenever this second OH, loses its oxygen, second OH loses its oxygen, this sugar becomes deoxyribose. And deoxyribose sugar, you will find it in the DNA, right? So you can clearly see the difference in both the sugars, right? Everything is same. The only difference is the second one doesn't have the OH. So this one is beta deoxyribose. It's a type of a derived sugar and you will find it in DNA. Which type of sugar is this? This is a derived sugar or derived monosaccharide. Right? I hope that's pretty clear to everyone. Yes, boys and girls. All right. Otherwise, we have also some other derived sugars like sugar acid. Like you must have heard of a NAG and NAM, N-acetylglucosamine, N-acetylmuramic acid. So all these also are derived sugars. What are these? These are also derived sugar. All right. So let's write the name of all the derived sugar. First category we have is deoxy sugar. That means the sugar which loses its oxygen. That's your deoxyribose that we have already discussed. Second is sugar acid. The sugar now contains some COOH group. You know, acidic group is COOH. So that becomes like glucuronic acid. If glucose have acidic group, right? Third is amine sugar, the one with NH2 group. Like your N-acetylglucosamine. N-acetylglucosamine. Alright. Sugar alcohol, the one with extra OH. That's mannitol. That's present in the fruits and in the algae. Right? Usually it's in the algae. So here are the mannitol, right? Mannose is the sugar. From there, extra OH added to the mannose, and that becomes mannitol. So that's a derived sugar that you will come across here. Like NAG will be present in your cell wall of bacteria. It will also be present in the arthropods, right? So for that, you have to make some suspense because we are going to learn that also. Okay, next is we have disaccharide. What are disaccharide? The one which have two sugars, right? So when two sugars join, they become disaccharide. So they must be bonded with a bond. So this bond is a glycosidic bond. So whenever a glycosidic bond forms, a dehydration process takes place. So there will always be a removal of water. In all the biomolecule chapter, whenever you have to form any bond, you have to remove water. Fine. So here, if you can see this sugar, this is your first, which is maltose. Which is the sugar? Maltose. Maltose is formed between two alpha glucose, right? as you can see here, this is the first carbon, second, third, fourth, fifth, and sixth. And this is alpha glucose. This is also alpha glucose. This is first, second, third, fourth, fifth, and sixth carbon of the another glucose. So first and fourth, they are forming this COC bond, because this is also C, this is also C. They had OH there, and those OH, they participate and they removed water, and the COC bond is alpha 1 4 glycosidic bond, because this bond is formed between first glucose 1 first carbon and another glucose fourth carbon. So this bond will be alpha 1 4. Let's see how. So if this is the glucose of the one sugar and this is the glucose of the another, right? They both have OH here. Because it's alpha, the OH will be down here, right? So they will be removing water like this. The water will be removed, and this type of a bond is formed. So glycosidic bond is actually the COC bond. Since it is formed between the first and fourth carbon, first carbon of first glucose and fourth carbon of the another glucose, you will name the bond as alpha, because we are using alpha glucose. 1 4 glycosidic bond. Alright, let's practice in them on the another disaccharide. Okay, the next disaccharide you can see is lactose here. Lactose is a milk sugar. Right? Maltose was a malt sugar. Why you call it as a malt sugar? From maltose. And where do you find it? You will find it in the germinating seeds, the seeds that are germinating and going to grow into a plant. There you will find the malt sugar. It is also part of base of number of health drink like Bonvitas and all. Then we have lactose, which is a milk sugar. It is formed of glucose, beta glucose and beta galactose, but the first one is always galactose. Beta galactose plus beta glucose. All right. So this sugar is beta galactose and this is glucose. Can you trace the difference between galactose and glucose? The only one difference, OH on this carbon, right? First, second, third, fourth, fifth, sixth. First, second, third, fourth, fifth, and sixth. The fourth carbon in the galactose has OH on the upper side. Here, if you can see in this, which ring we have drawn, all right, this. You can see in the fourth carbon of the glucose, the OH is on the lower side, right? So this is how it is. So here you can see the bond is formed between first and fourth, but the glucose used is beta. Then how will you name it? Beta 1 4 glycosidic bond. All right. Next we have is sucrose. Sucrose is formed between alpha, this is sucrose. Sucrose is your table sugar, the one you eat every day in the milk or something like that. It is formed between beta, you can see here, this one. All right, so first we'll write the glucose because this is glucose. Alpha glucose plus beta fructose. Alpha glucose or beta fructose. So this sugar is alpha glucose. This sugar is beta fructose. All right. So here we have first carbon of this sugar and here we have second carbon of this sugar. All right. The fructose, if this is fructose, this is the fructose, beta fructose, and this is the alpha fructose. So all the sugars that we have discussed till now, they were aldose. What were they? They were aldose, whereas fructose is a ketonic sugar. It's a ketose. So whenever a ketone group is formed, you need to have that group in between. That means CHO is aldehyde. So what's the ketone? You need to have something here and here. So that means the first carbon will not be the carbonyl carbon here. The second carbon will be the carbonyl carbon. Fine. So here the alpha glucose and beta fructose, they are forming a bond and the bond is formed between alpha 1 2, or you can also write it alpha 1 beta 2 glycosidic bond. Glycosidic bond. All right. So that's the three disaccharides that we have covered. Next, we are talking about the reducing and non-reducing sugar. So we have categorized the reducing and non-reducing sugar according to one experiment. And what is that? That's a Felling solution. What solution? Felling solution. We have one solution, the Felling solution. All right. We have what? We have Felling solution. So the sugar which can reduce the cupric ion into cuprous. The sugars that can reduce cupric ion of this solution into cuprous. These sugars, what are they known as? They are reducing sugar. What are they? They are reducing sugars. So we have two type of sugar, one are reducing, another are non-reducing. Reducing sugars are those, whenever you mix it in the Felling solution, they can turn this cupric ion in the solution to the cuprous. That means the solution is in blue color, as you can see there. When you add a reducing sugar, you will see the red color here, and that's a red one. So if I'm adding any sugar S in this solution and I get the red color, that means the sugar is a reducing sugar. If I add any sugar and I do not get the red color, that means the sugar is a non-reducing sugar. So which sugar are reducing? All the monosaccharide, right? Maltose, lactose. Whereas the non-reducing one is your sucrose. The non-reducing sugar is your, right? It cannot change the color. It cannot reduce the cupric ion into the cuprous ion. So if someone asks you which is a non-reducing sugar you have a sucrose. Fine. All right. Moving further to the next, we have polysaccharide. Poly means many. They have many sugars. They have many sugars. For example, we have glucose chain. We have a chain of glucose in a polysaccharide like this. All right. So this end will be the left end and this end will be the right end. So if someone asks you out of a left and the right end, which one is a reducing end, you will say the right end. What's the reason behind reducing and non-reducing? Why the reducing can change the color and non-reducing cannot? Because if you see, all these sugar have free functional group. And what is free functional group? Their aldehyde. Fine. But if you see this one, you can see alpha glucose have first carbon as a carbonyl carbon and carbonyl carbon have the functional group. So it has been utilized in the formation of glycosidic bond. Similarly, the fructose will be having the carbonyl carbon or the functional group at the carbon 2. So both carbonyl carbon and the functional group has been utilized in the formation of glycosidic bond. So there is no other functional group present because all these are hydroxyl. They are not aldehyde or ketone. They cannot reduce the cupric into cupric. Fine. So for that you need to have the free aldehyde group. So if I have a chain of polysaccharide, this one will not be having any functional group. The right end will be reducing because it contain functional group. What is a functional group? Like aldehyde. Because it's the glucose. Alright. We have two type of polysaccharide. One is homopolysaccharide and another is heteropolysaccharide. What's the difference? Homo means similar and hetero means different. Hetero means different. Fine. Homopolysaccharides are the one which have same units, right? If it is made up of glucose, everything will be made up of a glucose. So they have same sugars. Whereas they have different sugar. Like starch is a homopolysaccharide. It is just made up of all the glucose. Whereas, all right, whereas the cell wall of bacteria, it contains two types of sugar. So it's a heteropolysaccharide. All right. Also, your other type of the sugars are also there, but I'm just giving you one examples. So wherever the word homopolysaccharide comes, just you have to remember it will be containing of only one type of a sugar, whereas hetero means it will be containing of different types of sugar. Let's talk about polysaccharide. In polysaccharide, first we have starch. The starch is formed of two components. If you look around the starch, you will see two type of structure. One is amylose, another is amylopectin. The starch is made up of alpha glucose. What it is made up of? Alpha glucose. The amylose is helical structure, and this one is a branch structure. So what you are seeing there is a branch structure. Now what is a branch structure? If you say this is a straight line, and out of this straight line a branch is coming out. Can you see that? Simple. So in a straight line, all are forming alpha 1,4 glycosidic bond. So if I say it is unbranched and it's helical like this, helical means like this, it will be having alpha 1,4 glycosidic bonds everywhere. If amylopectin is this structure, then in the straight chain, amylopectin will be having alpha 1,4 glycosidic bond, whereas in the branching, it will be having alpha 1,6 glycosidic bond. Can you see that? Here we have fourth carbon, here we have fifth, then here we have sixth. So its first is forming bond with its sixth carbon. So that's why at the branching, sixth and first are participating in the formation of glycosidic bond. So here at the branching, the bonding is alpha 1,6 and here it is alpha 1,4. Do not forget that. And you also must have heard of that the starch gives iodine test, right? Starch gives blue black color with iodine. Have you ever heard of it? Yes. So starch gives blue black color with iodine. The reason is because amylose have helical strand which traps the iodine. So starch gives blue black color with iodine, right? Amylose traps the iodine. So where is the starch formed? It is present in plants. And how do the plants form the starch? They form it during photosynthesis. All right. Moving further to next, storage one. Okay, so if you must have read this word, the storage polysaccharide. The starch is a storage polysaccharide. Why? Because it gets stored in the plants. Similarly, we animals also have a storage polysaccharide. And what's that? That's our glycogen. So where do you find glycogen? In muscles and liver. In animals, we will see the glycogen, which is a storage polysaccharide. And where in the animals? In the liver and muscles. It is also made up of alpha glucose. It is also a branched structure, right? So it's not just like it's only present in animals. Yes, it is not present in plants for sure, but in bacteria you can also see the glycogen granules. So apart from animals, it is also present in bacteria. Okay. Since it's a branch structure, its structure will exactly be like that of amylopectin. As you can see, this straight chain. Yes. Can you see the straight chain? In the straight chain, the bonding is alpha 1,4. Whereas the branching occur, or just look at this diagram. In the straight chain, you will see alpha 1,4. And wherever the branching occur, it will be alpha 1,6. Exactly like that of amylopectin, right? So straight chain, the bonding is alpha 1,4, and at branch It will be performing alpha 1 6, or it will be forming the glycosidic bond with the first carbon of one sugar and 6 carbon of the another sugar. Next in storage, we have inulin. Inulin is made up of fructose. It's a fructose polymer. And where do you find it? It's again a storage polysaccharide of plants like artichoke and dahlia. In human, this inulin, first of all, it's inulin. It's not insulin. Insulin is different and it's proteinaceous. This is inulin, which is a storage polysaccharide of artichoke and dahlia, and it is made up of fructose sugar. And it is used to check kidney function. So, where do we use it? Whenever we have to see whether the kidney is functioning properly or not, we perform this test, right? So, a person is injected with inulin. If the same amount of inulin gets excreted in the urine, that means the kidney is functionally performing well. Because in our body, the fructose is not metabolized, and whatever thing is not metabolized in your body, the kidney excrete it out, right? So that's why it is used to check the kidney function. Next, we have the structural polysaccharide, that's cellulose. That's really very important. Cellulose, where it is present? It is present in the plants. It is present in the plants, and it is made up of beta glucose, first of all, right? It is also homopolysaccharide. Whatever we have discussed till now, all were homopolysaccharide, all were made up of one type of sugar. Now, a lot of students get confused. Ma'am, in starch we have amylose and amylopectin. These are the various type of structure, but or the criteria to identify homo and polysaccharide is the type of sugar. It has only one type of sugar, so that's homopolysaccharide. So it's a structural polysaccharide. Why? Because it is present in the plant cell wall. It is forming some structure. What it is made up of? It is made up of beta glucose, and we cannot digest it. Why? Because we have alpha salivary amylase, or we have all the enzyme in the digestion which can digest alpha bonds, not the beta bonds. That's why you cannot digest cellulose. Okay, that's why whatever food you eat raw, like cucumbers, if you eat raw spinach, or you eat raw capsicum, that's all are a part of roughage, or your salads. They actually are known as roughage because they are not digested in your body, because they have cellulose. The cellulose is intact there. Whenever you cook the food, during heating process, the cellulose get disintegrated, and now you can digest the food, the internal part of the food. But whenever you eat raw, that becomes a part of a roughage. You cannot digest it, and it moves out in the feces. And hence roughage or salads, they prevent constipation, because then they will become a part of feces. Okay, then here the cellulose, you can see it's unbranched. It's unbranched. Whereas glycogen was branched, starch was branched, this one is unbranched. It's the straight chain. There are a lot of uses of cellulose. First of all, it's the most abundant organic compound. It's the most abundant organic compound. Most abundant organic compound. Next, uses. First, it is used to make paper. It is used to make artificial silk, and that's rayon. Rayon. The natural silk is a protein, whereas artificial silk is a rayon. Also, it is used to make explosives like bombs, right? Or bullets. Fine. Otherwise, it is also used as a solvent in medicines or cosmetics and all. Fine. So there are a lot of uses of cellulose. Some of them we have discussed. All right. And as you know, it's only made up of beta glucose, so it's the homopolysaccharide. Next, structural polysaccharide. Again, in storage we have done starch, glycogen, and inulin. In the structural, we have done cellulose. Now that's next one is chitin. Chitin is present in the exoskeleton of arthropod. So, skeleton of arthropod. Alright, arthropod like insect, like your favorite cockroach. Fine. So, what it is made up of? It is made up of NAG. What is NAG? N acetyl glucose. So, it's a modified sugar. NAG is also present in the cell wall of bacteria. And the bonding is beta, and it's the unbranched structure that you don't need to actually learn about, then it will become more complicated. The only thing that you need to remember is it's the present in the exoskeleton of arthropod. And also, if I talk about botany, it is also present in the fungi, in the cell wall of fungi. Okay, all right. Next, let's solve some questions. First, which of the following is not present in the acid insoluble fraction? What was acid insoluble fraction? The one which was retentate. And in the retentate, we had macromolecules and lipids. So we have to tell which is the one which is not present in the macromolecule, or which is not the macromolecule, or which was not present in retentate. Protein was in retentate, nucleic acid and lipid, but glucose, since it's a monomer, it was present in filtrate. So answer is fourth. Next question. Cellulose is a heteropolymer made up of glucose and galactose? No, it was a homopolymer of only glucose, not galactose. Homopolymer made up of alpha glucose? No, homopolymer made up of beta glucose. So it was not a heteropolymer. So rule out the heteropolymer because it was a homopolymer of what? The beta glucose. So answer is third. Alright, inulin is a polymer of, inulin is a structural polysaccharide of Etrichok, which was a polymer of fructose. So answer is 2. Match the following column 1 and 2. So it's a match the following of the secondary metabolites. Alkaloid. So alkalo was morphine and codeine. So we have codeine here. Lactine viscose concanvalin A. Drugs we have curcumin, the one in the turmeric, and toxin we have ricin. So you need to learn all of this. Moving further to the next is your amino acids. Let's talk about much interesting thing, amino acids. Alright, so let's talk about amino acid. As the name suggests, they will be having amine group and acid. They are actually substituted methane. What is a methane? CH4. So carbon with 4 groups. And all these groups will be different. One is like hydrogen, another R group. R group is different in different amino acid. One is COH, the acidic group, and the amine. So this carbon is containing all the groups on this one. So this carbon is alpha carbon. Since we have alpha carbon in the amino acid, all amino acid are alpha amino. Since we have all the groups that are different, so these type of structures are asymmetrical. So we say the alpha amino acids, they are asymmetrical. What is asymmetrical? All groups different. But since we know the biology is the field of exception, but there is again one exception, except glycine. What is different in glycine? Glycine is symmetrical. Because if any of the group matches, the compound definitely becomes symmetrical. So what's the structure of glycine? We have carbon, and then we have hydrogen and hydrogen. Then we have COH here, and then we have NH2, right? This above 3 structure is same in all the amino acid. The only different group is the R group. In glycine, we have H as R. R group is different. So it can be H, it can be CH3 in some, it can be CH2, CH3, anything can be there. So here in glycine, we have R group, that is H. So both these are same. So we say glycine is symmetrical, right? How many protein forming amino acids do we have? We have 20 protein forming amino acid. Otherwise, we have a lot of amino acids. But if someone asks you protein forming amino acid, you will straight away say 20 amino acids. How many? You will say 20 amino acids are protein forming amino. Fine. Okay, let's talk about the amino acids in more detail. So the thing which we are going to discuss is zwitterine. So we say these amino acids, they work or behave differently in a different solution, and that depends upon the pH of a solution. So what type of a pH is that solution is having, that amino acid will behave as anion or cation. All right. So if I say I have this amino acid, if I say I have this amino acid, and in this amino acid, this is acting as an ionic form. Fine. This is acting as an anionic form, right? So this is like pH X. At HPX, the amino acid act as anion. Fine. Also, there can be another pH where it can act as a cation. Fine. So according to pH of a solution, the amino acid can act as an anion or it can act as a cation. But in some cases, it can also act as in the both forms. That means anion as well as cation. And you call it as a dipolar state or a hybrid state. And the another name for hybrid is zwitterine. So what is a zwitterine? It is one form or ionic form of the amino acid at a particular pH, and you call it as isoelectric point. So at isoelectric point, amino acid behaves as dipolarine, and that stage is known as zwitterine. Zwitter means hybrid. All right. So if there is a type of pH, it can act as cation or anion or a dipolar ion. So it all depends upon the pH. But there is one point known as isoelectric point where it acts as a Zwitter ion form. It can be different for different amino acids. So that particular pH, what do you call it as isoelectric point, is different for different amino acid. That depends upon what type of R group an amino acid is carrying. So let's move to that then. Classification of amino acids. So the first classification is according to structure. I hope you can see it clearly, right? The first classification is according to structure. What do you see in structure? You see in structure the R group. What do you see in structure? You see in structure the R group. If I talk about R group, this R group can be, neutral. It can be acidic. It can be, basic. Any type of R group can be there. For example, if I talk about glycine, it has hydrogen. Alanine, it has CH3. Valine, it has CH, CH3, CH3. And leucine, again, carbon, 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 isoleucine, carbon, carbon, carbon. All these three, they are having a neutral. They have having neutral groups. So if I talk about any other, like if I talk about here the uh, the one with the negative charge, like aspartic acid, you can see here the group is CO negative is present. So that means it is having some charge. If something is not having charge, you call it as a non-polar one, right? Again, non-polar and neutral are different. In neutral, there is no polarities there, no charges there. In non-polar, maybe the charge is there, but that has been neutralized. So that's the non-polar. Okay, that means if I have two negative charge, I have two positive charge. That means I have charge, but there is no polarity. That's non-polar. Second thing, if I have no charge, at all, the group is electrically neutral. So according to our group, the first category that we have is of neutral amino acid. We have neutral amino acid. In neutral amino acid, we have GAVIL, GAVIL. So here is a one letter abbreviation written: glycine GAVIL, alanine A. Glycine, for glycine we have G, for alanine we have A, for valine we have V, leucine L, and isoleucine I. So that becomes GAVIL. Alright. Now the classification which we are doing is according to the structure. So don't focus upon this charge, positive, negative, just don't focus at all. Fine. Then second classification is of the acidic amino acid. The acidic are the one with the negative charge here, right? So all these are acidic amino acid. What are acidic amino acid? Aspartic acid and glutamic acid. One thing that you will notice, this structure is same in all the amino acid. The only difference is the R group. Here you have CH2COH. Here you have CH2CH2COH. Fine. Next, the one with the positive charge are the basic amino acid, are the basic. You can see here the basic. In basic, we have lysine, arginine, and histidine. Lysine, arginine, and histidine, right? Histidine also comes in the category of heterocyclic. Which one? Heterocyclic. So if I'm talking about the positive charge, all these three. If I'm talking about basic amino acid, I will take only lysine and arginine. What I'll be taking? Lysine and arginine. Then next category of the one which contains sulfur. The one which contains sulfur are two, methionine and cysteine. So these are the one which are sulfur containing. So here in the R group you can see sulfur, in the methionine and cysteine CH. So the difference is CH2, CH2, SCH3. Here CH2 and SH. Next comes the category of the aromatic one. In aromatic one, you should have a benzene ring. So which are the one with the benzene ring? Phenylalanine and tryptophan. And also your tyrosine. So these three, these are the aromatic one. These are aromatic. Alright. Moving further to the next one. The one with OH group or alcoholic. So the alcoholic are these one. Alcoholic are these one. OH group. Serine and threonine. Serine and fine. And here we have asparagine and glutamine. These are derived amino acid, right? So they have been derived from aspartic acid and glutamic acid. All right. And then comes the heterocyclic. For heterocyclic, we have histidine and proline. Fine. So this is for you so that you can see the structures of everything. The most important structures are of some of the amino acids. So I'm putting the star in the pink, the structures you have to do. All right. So here we have glycine, alanine, valine. These are the must structures that you have to do. All right. Otherwise, you have to do tyrosine. You also have to do the glutamic acid. In any course, you have to learn these structures. All right. So if this becomes a bit difficult for you, let me simplify this for you. So if I'm talking about according to our group, the first are neutral amino acid. In neutral amino acid, we have usually the chain of carbons, right? Here you will be having one NH2 group, one COH group, and one with the CH2 or CH3. So here comes the gavel. Glycine, alanine, valine, isoleucine, and leucine. Second classification is the acidic one. In acidic, you have another acidic group. So here becomes one NH2 group and two CO. And here we have aspartic acid and glutamic acid. All right. Then moving further to the next, we have basic amino acid. In basic, what will be the extra? NH2, because amine gives the basic group, right? Amine is a basic group and acidic is the COH. So here, what do we have? 2 NH2, 1. So what is here? In the basic, we have lysine and we have arginine. Moving further to the next, sulfur containing. In sulfur containing, you will be having sulfur group. We have methionine and cysteine. Then we have alcoholic, the one with the alcohol group, serine and threonine. Serine. Okay. Likewise, we have the aromatic ones, right? We have aromatic ones. What are the aromatic ones? In aromatic ones, we have the tyrosine tryptophan, and phenylalanine. Here you will be having a benzene ring. Okay, so it's quite simple to identify their structure because they are not going to ask you for very high standard. For example, they gave you one acidic, one neutral, one basic, one sulfur containing, right? And they ask you which of the following is acidic amino acid, right? You just have to simply see which one is having the extra COH. And that's simple. Likewise, if they gave you the structure of basic and they ask you which of the following is a basic amino acid, because in basic you have two NH2, so these type of questions will arrive in the exam. Nobody is going to ask you draw the structure of the glutamine. No one. No one is going to ask, right? Okay. Then the last category, heterocyclic. Okay. In heterocyclic, we have heterocyclic rings. Now, what are heterocyclic rings? These are the rings which are not just made up of carbon, but they have other compounds as well, like nitrogen, right? So here we have proline, histidine. Fine. So these are 18. So other two are the modified versions. They are like glutamine and asparagine. If I talk about charge, so we have non-polar amino acid, we have polar amino acid, and then we have polar charge one, right? So this is how the classification occurs according to polarity. So for that you have to see, just because acidic one have charge, the basic one will always be having charge. So all the acidic and basics are the positively charged one. The one which have the charge, but they are polar, but they are electrically neutral. So these are serine, thurine, tyrosine, asparagine, and glutamine. Others all are non-polar. Fine. So according to charge, we have these type of amino acid. Next classification is according to how much important are they for your body. We have essential, semi-essential, and non-essential. Essential. What are essential amino acids? These are those amino acids which are not produced in your body. You have to, eat it from the external diet. So they are very much important. For example, there are certain food which you cannot, you cannot cook them in your kitchen or at your home because they are so cumbersome, or you don't know how to cook them. Okay, or you don't have all the ingredients to cook them. So what do? You just go to the restaurant or you ask the delivery guy to get it for you. Fine. So they are so much good for you. For example, you cannot make a pizza like other pizza brands make. Fine. So for you, that's the most yummy thing, right? But if I talk about any sambar you make at home, or salan, or any dal which your mom cook at home, you will say, oh, that's just that dal or that sambar. Fine. But if we think about another thing that's so you crave about that thing, because that's not produced in your home, so you have more importance for that thing in your life. Fine. So the essential amino acids are those amino acids which you cannot produce, your body cannot produce. So you have to take it from the external diet. And these are all these. You can see these. So, but we'll make it easy, don't worry. So essential amino acids are those. These are not produced in our body. In our body. So these are lysine, leucine, isoleucine, valine, phenylalanine, methionine, tryptophan, threonine. Right? Then we have semi-essential one. Semi-essential one are arginine and histidine. These are the one which are produced in a body but in a very less quantity. So these are produced in very few quantities. So again you have to take it from external diet. Whereas rest which are left, because we have 20 total, so what are left, they can be produced in a body, so they are non-essential. So these can be produced in a body. All right. So how do you learn it? So you can learn it by live life in Vegas. You all know what is Vegas, right? That one in America. So live life in Vegas must try protein tea, right? Live life in Vegas must try protein tea. Okay, so you can just shift protein here. So I can even write it here for you. Live life in Vegas must try protein tea. Okay, so like this you can learn it actually. Okay, so this is the classification according to nutritional requirement. That means how much nutrition do you need. I means whether you need it or not, whether the amino acid so much important for you or not. Fine. Okay, next is your functions of amino acids. So what are the various functions performed by the amino acid? The first thing is amino acids forms proteins. They forms protein. Some amino acid like tyrosine, it forms thyroxine hormone and adrenaline hormone. Fine. They also produce neurotransmitters. What are neurotransmitter? These are chemicals that we have discussed in the nervous tissue. Okay. These are the chemicals that are secreted in synapse. All right. Moving further to next, what are the other functions? They also form heme of hemoglobin. They form heme. All right. Okay, so these are the some of the functions of the amino acid. Moving further to the proteins. So what are proteins? These are the heteropolymer. Hetero means different. Heteropolymers of amino acid. What is a heteropolymer? That means it will be containing a different type of amino acid. For example, glycine, alanine, valine, isoleucine, leucine. I would be calling it homo if all were glycine, if all were alanine, or if all were valine. Right? So, but here different types of amino acids are used, so you call it as the heteropolymer of amino acid, which is the most abundant protein in the animal world. In animals, most abundant is collagen. Whereas in the biosphere, in the entire biosphere, most abundant is your rubisco, which is used in the plants. Yes, photosynthesis. All right. So, how do they look like, or how these amino acid forms the protein? So, for that, you need to understand the structure of a protein. So, there are different levels of structure of a protein. Like we have primary structure, secondary structure, tertiary, and quaternary. So, this is a way to write them, another way to write them. The primary structure is just the long chain of amino acid, right? Just one after the another. One, two, three, four, like this. When this straight chain, for example, if you just make chain, if this chain, it looks like that, and that's the molecular structure. When this chain, it gets coiled up, right? Or it gets in the form of plates, and hydrogen bond stabilizes it. That's your secondary structure. So secondary structure can be of different type of shape. It can be in the form of plate, that's beta pleated, or it can be in the form of alpha helical structure. If this one gets coiled more and become the size of a woolen ball, then it is your tertiary. And if these four balls, these are packed together, and forms a single protein, you call it as a quaternary structure, right? I have a single wire, that's this primary. I coiled the wire, that's the secondary structure. If I, push a lot, give lot forces and make a woolen ball, that's the tertiary. If I have these four balls packed together, that's the quaternary structure. Fine. All right. Okay. So let's talk about them in little detail. So if I'm talking about the primary structure, how is it formed? I have this amino acid with COH group here, NH2 and H and R. Fine. Then here another amino acid will come and will try to make bond. And I've told you in the polysaccharide as well, whenever the bond is formed, there is removal of water. There is removal of water. So this bond formed will be CONH. And you call this bond as not the James bond, peptide bond. Which bond is this? This bond is the peptide. Okay. So in a primary structure, this is a primary structure. Which bond is present? Peptide bond formed by the removal of water. It's a type of amide bond. What is amide? Because it contains NH2 or NH. It's a type of covalent bond. Here NH2 is free, so this is N-terminal. And here COH is free, so this is C-terminal. So whenever you have to read the sequence of amino acid, you need to know the primary structure first of all. And you always read from N to C. That means this one will be the first amino acid, this one will be the second one. We always read from N to C. Okay. So that's the protein's primary structure. And whenever you have to read the sequence, you use the structure. If I talk about the secondary structure, which is alpha helical and beta pleated, let's talk about them. Alpha helical structure. In this, the helix is always right-handed, like this. Right? It is right hand. Here, the strand is stabilized by which bonding? Hydrogen bonding. Right? And hydrogen bonding is a bonding that takes place between one hydrogen atom and one highly electronegative atom. So here the highly electronegative atom is OH. So here hydrogen bonding stabilizes it. If someone asks you which bond stabilizes the secondary structure, what will you say? You will say hydrogen bond. Where do you find it? You will find it in the myosin, tropomyosin that we will study in the muscular part. Okay, so here you can find them, the alpha helical structure. Talking about the beta pleated. In beta pleated, we have different pleats like this, one upon the other. If this is N, this is C, like this. Okay, this is also stabilized by hydrogen bond. Okay, hydrogen bond. All right, this one, the alpha helical structure, you can also find this in the keratin. What is keratin? Keratin is a protein. Keratin is a protein you find in the nails and all. And this structure is also exhibited by collagen. Collagen also exhibited this structure. All right. Okay, so where do you find the beta pleated? Beta pleated can be found in the keratin of insects and also in the silk protein. Also in the silk protein. So here the arrangement of these fibers can be parallel as well as antiparallel. So the beta carotene of the animals, they exhibit the parallel fashion of these, like one upon the other, whereas the silk protein will show the anti-parallel beta sheet like this. Like this. Okay, so there is a difference. Alright, so this one is parallel N to C, N to C. Whereas in anti-parallel, there is different C to N, N to C, like this. Okay, all right. Moving further to the quaternary structure, tertiary structure. You can see here in tertiary structure, hydrogen disulfide bonds are written, right? So tertiary structure is a woolen ball structure. Here, a lots of bonds are there to stabilize it. What are these? Hydrogen bond, these disulfide bonds, ionic bond, van der Waals forces. All these are there to stabilize this structure. All right. Sometimes excessive heat can actually disrupt the structure, and you call it as denaturation. So, where do you find this tertiary structure? We will find this tertiary structure in the enzymes. And where do you find this quaternary structure? Where there are different subunits. So, all these are subunits. There are 4 subunits. You will find this in your hemoglobin. Okay. So if someone asks you hemoglobin exhibit primary, secondary, tertiary structure, you will say the quaternary structure. All right. And enzymes, the tertiary structure. All right. Moving further to the classification of protein. Proteins are of two type. Proteins are of two type. One are simple protein and another are conjugated. What's the difference? In simple protein, you just have the sequence of amino acid like this. Only protein is present, simple. But here, along with this chain, you will find some prosthetic group attached. And this prosthetic group can be anything. This prosthetic group, it can be a carbohydrate, it can be a lipid, it can be a nucleic acid, it can be anything. Fine. For example, your antibodies, these are actually the protein with carbohydrate. So this category of protein is known as glycoprotein. Okay. Chylomicron, which is produced in your digestive system, that contains lipid. So that is a category of lipoproteins. All right. So this is how you have different types of proteins. Next is function of proteins. So what are the functions a protein performs? First, collagen. Collagen is a part of intracellular ground substance. That one we have done the matrix in the connective tissue. Trypsin act as an enzyme. It catalyzes a lot of digestive chemical reactions in your stomach, or if I talk about trypsin, in the small intestine. Insulin, it's a hormone. Antibody, it fights infection, it's a glycoprotein. And you know, whenever you get infection, so your body's immune system, it produces these glycoproteins to kill the microorganism. Then we have receptor like sensory reception, smell, taste, hormone, etc. Like in nose, we have olfactory sensory receptor that picks up the chemical and you get to know the smell. GLUT4 enables glucose transport into cell. So GLUT4 is a type of transporter present in cells. Fine. If this is a cell, this is GLUT4. It will allow the glucose to get into the cell. It will allow the glucose to enter into the cell or go out of a cell. So that's why they have mentioned the word transport. Fine. Alright, so these are the various function of protein. Let's talk about the lipids now. Okay, so before lipid, let's solve some question. Hydroxymethyl is the R group in amino acid glycine, alanine, serine, aspartic acid. So what is hydroxymethyl? CH2OH. Now, if you know this is CH2OH, hydroxyl OH, and you know OH, the one which contain OH group are serine and threonine, you can simply answer this question without reading all the option. Glycine contains H as R group, alanine contains CH3, CH contain OH, and this one contains extra COH group, right? So straight away the answer is 3, which is serine. Fine. Next, which of the following is an essential amino acid? Arginine is your arginine and histidine, both are semi-essential. right? Let's move here. So here we have done live life in Vegas. Live life in Vegas, must try protein tea. There was no G, right? Live life in Vegas. There was no G mnemonic, so glycine is not the essential. So lysine is the essential. So answer is 4. So you can, if you have learned the mnemonic, you can easily crack it. Next. Choose and correct statement from the following. Amino acids are substituted methane. Absolutely correct, because these are like methane, the methane which have different types of group. In protein, only right-handed helices are observed, right? Tertiary structure of protein is absolutely necessary for the many biological activities of protein. As I've told you, if there is, excessive heat, the protein structure can get denatured, right? It can get destroyed. So, well, if it can get destroyed, its biological functions will be disrupted. For example, I have given you example of enzyme. If the tertiary structure of a protein gets denatured, or if you open up the tertiary structure, will enzyme be as intact as it was be? No, it wasn't. The enzyme actually gets broken up. If enzyme get broken up, will it be catalyzing the reactions? No. So that's how we say the tertiary structure is used for the biological activities of a protein. Fine. Whereas primary structure help us to read the protein sequence or amino acid sequence. So this is also correct. In a polypeptide or a protein, amino acids are linked by a peptide bond which is formed when a carboxylic group reacts with the amino acid group of the same amino acid. No, the carboxylic group and NH2 group, they are of the different amino acid, not the same amino acid. So answer is 4. You have to find which one? Incorrect one. Okay. All right. Next. So next we have lipids. Let's talk about lipids. If I talk about lipid, guys, lipids are water insoluble. What are they? They are water insoluble. You cannot dissolve them in water like oil. If you get a drop of refined oil or any oil on water, does it dissolve? No, it's water insoluble. But they can be solubilized in other non-solvent uh, solutions like ether and benzene and all, right? So lipids. Now what is a lipid? A lipid can be a single fatty acid. A single fatty acid can be a lipid, or fatty acid plus alcohol can be a lipid. And usually this alcohol is a glycerol. So any of these can be known as a lipid. So as I've told you, fatty acid. Now what is a fatty acid and what is a glycerol? First of all, glycerol is trihydroxypropane. What is propane? The one with the 3 carbon. And I've told you trihydroxy. That means it will be having 3 OH. So trihydroxypropane is your glycerol. So whenever glycerol or trihydroxypropane, it combines with fatty acid, it forms the ester bond and the process is esterification. So whenever they combine, when acid and alcohol combine, they form ester. Fine. And the process is esterification and the bond is ester bond in between these two. Fine. Now talk about fatty acid. What is a fatty acid? Fatty acids are actually long chain acid. Okay. Here we have carboxylic acid with long R chain. So they are particularly also written as RC. This R can be different. For example, we have this palmitic acid. It has 14 and 15 and 16 carbon. How many carbon does palmitic acid have? 16. How? 14, 15, 16. If I exclude carboxylic carbon, then it will be having 15. There are two types of fatty acid. One is saturated, another is unsaturated. The saturated one are the fatty acid which have single bonds, and these are the one with the double bonds, unsaturated. They have high melting point, they have low. So whenever you talk about or you think about saturated, think about ghee. So ghee in winters, it freezes, right? And that's why we say it has high melting point. It needs more temperature, or it needs more temperature to melt up, right? Then here unsaturated, saturated, they have low melting point. What are the example? Palmitic, steric, and ericatic. They are all saturated. That is without double bonds. Olic, linoleic, linolenic, arachidonic. They are what? They are the one with the double bond. So here, in the oleic acid, we have 18 carbons. In linoleic and linolenic, again we have 18 carbons. Now, what is this 1, 2, and 3? These are double bonds. In oleic, we have only one double bond, and here 2, 3, and in arachidonic, we have 20 carbons and 4 double bonds. Okay, so that's why you call these as PUFA. What is PUFA? Polyunsaturated fatty acid. More the bonds, more the bonds, more healthier the fat is, right? So these are so good to eat. They are present in olive oil, gingerly oil, so these are like good ones. In here, palmitic, we have 16 carbons, steric 18, and then 20. All these are including carboxylic carbon. If I say, tell me the number of carbons present in steric acid, you will say 18. If I ask tell me the number of carbons in steric acid without COH or excluding COH carbon, you will say 17. Okay, so these are the various fatty acids. All right, you have to memorize the structure of palmitic acid for sure, right, guys? Okay, then classification. What type of lipids do we have? We have three type of lipids. One is a simple lipid with no complication at all. That means it will be formed of a fatty acid and alcohol. Then we have compound. It will be containing another groups, and derived is altogether a different world. Write this down. In simple lipids, we have fatty acid plus alcohol. In compound, fatty acid plus alcohol plus one another group, right? Derived, different category. Fatty acid and alcohol are one simple lipids. They contain true fats. They contain waxes. Now, waxes are of different type. Like we have ear wax in human. We have plant wax. We have bee wax, the one from honeybee. These all are what? These are simple lipids. In compound lipid, we have phospholipid, right? We also have lipoprotein, which we have already discussed, chylomicron. Fine. We have glycolipid. In derived, we have steroids. We have, all right, so let's just talk about steroids here. Okay. So here, if I talk about the simple lipids, so the most common is a true fat, The true fat is made up of glycerol. We have glycerol here. We have glycerol here. So when you want to esterify this glycerol with fatty acid, what you need? Obviously a fatty acid. So if I have this fatty acid, and as I've told you, whenever I have to make a bond in biomolecule, I'll be doing, I'll be removing water only. So let's remove water as always. HOH. I have removed the water. Now what I will get? I will get a monoglyceride. I will get a monoglyceride because this one is having only one ester bond. So this product is known as a monoglyceride. If I add another fatty acid in the monoglyceride, what I'll be having? I will be having diglyceride. I will be having diglyceride. Okay, so here comes the, all right. diglyceride. So if I have to make the triglyceride, you know what to do. Add another fatty acid. Okay, so I have added another fatty acid R3. So what I'll get? I'll get the triglyceride. Okay, so if someone asks you the triglyceride is formed of, what will you say? You will say it is formed of one glycerol. This is one glycerol. Yes. This entire structure is one glycerol. Okay. One glycerol and how many fatty acid? Three. So one triglyceride is formed of one glycerol and three fatty acids. So just make sure you should not confuse. This is the entire structure of glycerol. It has three OH. That's why it can is esterified with three different fatty acids. All right. Moving further to next. Next we have, okay, compound lipid. In compound lipid, we will discuss the phospholipid. So as I've told you, the lipid with another group is a phospholipid. So that means it will be having glycerol, will be having the fatty acid, plus it will be having another extra group. So here in this case, phosphate. Here what will be having? Fatty acid, glycerol, plus. Okay. So here we are discussing the structure of lecithin. Lecithin is present in the cell membrane. Where it is present? It is present in cell membrane and especially in the alveoli. So this is the structure of lecithin. Lecithin is having a nitrogenous, is known as choline. So since it is phospholipid is made up of fatty acid plus glycerol plus phosphate, let's search. So this is glycerol. These are fatty acid. What are these? These are fatty acid, glycerol. Okay, and then we have this phosphate, and here another group which is nitrogen. So you have to just have a pictographic memory of the structure, because sometimes they directly draw the structure there in the diagram, and they ask you which structure is this, and if you know that's the structure of lecithin, the all marks are yours. Fine. Okay, so that's the compound lipids. In compound we have discussed the phospholipid. Talking about derived lipids. In derived lipids we are going to talk about steroids. First of all, what's wrong with the derived lipids? Nothing is wrong with derived lipid. They are actually lipid that have been derived from the normal fatty acid or glycerol. They are not generally fatty acid plus glycerol esterifications. They are derived from fatty acids. They are derived from fatty acids. So here the first is steroid. In steroid, the most common is sterol. The most common is sterol. Sterol are the steroid with alcohol group with OH. So cholesterol is one example of a sterol. And this cholesterol is exclusively present in animals. Where it is present? Animals. So you can see this four ring structure of cholesterol. So what is the role of cholesterol in our body? First of all, it's present in your cell membrane. Where it is present? It is present in our cell membrane. Second, it forms our sex hormones like estrogen, progesterone, testosterone is formed by cholesterol. It also produces bile and vitamin D. Yes, vitamin D is produced in our body on exposure to sun rays. So vitamin D gives the precursor of the cholesterol gives the precursor of vitamin D, and then on exposure to sun ray, the reaction gets triggered and it gets converted into vitamin. Bile we will study it in the digestion, that bile helps in the emulsification of the fat. All right, so that's about the lipid, guys. Now we'll solve some question. Dash has 16 carbons including carboxyl carbon. Very straightforward. Palmitic acid. Arachidonic has 20 carbons. Arachidic has 20 carbons. Oleic has 18 carbon. So answer is 1. Next, lecithin is a glycolipid, phospholipid, sterol, and simple lipid. You know, lecithin is a conjugated lipid with phosphate group in it, so it's a phospholipid. So that type of a question can arise, right? What about glycolipid? Glycolipids are also type of lipid with carbohydrate in them. They have carbohydrate. Like in phospholipid, we have phosphate, they have carbohydrate. So, where do you find them? You find them in your neurons or nerve membrane. Then we have simple lipid. That's not the answer. Next question. Identify the structure and find the correct statement. So, here you can see the structure. This is the structure of triglyceride. And you have to find the correct. Triglyceride made up of 3 glycerol? No. Triglyceride made up of 1 glycerol and 3 fatty acid, but here is diglyceride. So wrong. Triglyceride with 1 glycerol and 3 fatty acids. So answer is all right. Okay, so let's move further and talk about nucleic acid. All right, so let's talk about nucleic acid. So if I talk about the nucleic acid, you have two nucleic acid in your body. One is DNA and another is RNA. These nucleic acid, they are heteropolymers, hetero again, different types of units, heteropolymers of nucleotides, right? What is a nucleotide? Nucleotide is a nucleoside plus phosphate. Now, what is a nucleoside? Nucleoside is sugar plus nitrogenous base. So if someone asks you what are the components a nucleotide is made up of, we will say it is made up of sugar, nitrogenous base and phosphate. Let's talk about these components in detail. So here we have these sugar. One is a ribose, another is a deoxyribose. Now you have to identify. I give you 10-second time because we have already done this. You have to identify which sugar is this. Okay, I have told you one is ribose, another is deoxyribose. Which one is deoxyribose? The one which do not have O at the second carbon. So this one is deoxyribose sugar. So one component of nucleotide is sugar. It can be deoxyribose, it can be ribose. If I talk about DNA, in DNA you will find deoxyribose, whereas in the RNA or ATP or other nucleotides, free nucleotides like FMN, FAD, in all these you will find the ribose sugar. Here we have phosphate group or phosphoric acid. It depends upon what type of ionic form it is present. And then guys, then we have the third component, which is the nitrogenous base. Now what are these nitrogenous bases? These things must be new to you. Nitrogenous bases are heterocyclic compounds. Again, heterocyclic we have discussed in the protein part amino acid. Heterocyclic means the cyclic or the chain or the ring, not the chain, the ring that is made up of carbon as well as nitrogen. As you can see here, one nitrogenous base is adenine. So here you can see the chain is made up of carbon. So these sides are carbon, carbon, carbon. Along with it, nitrogen is also present. So the rings in which nitrogen is also present along with the carbon, you call them as heterocyclic. So here we have two types of nitrogenous bases. One are purine, another are pyrimidine. All right. So purine, we have two types of purines. You can see one is their adenine. The purines are usually double cyclic. That means it has two rings. Whereas pyrimidine, the one which is present here, uracil, these are single cyclic. They have only one ring. In purine, we have adenine and usually it is denoted with A, and we have guanine, which is G. Then in pyrimidine, we have cytosine, which is C, uracil, which is U, and thymine, which is T. So here there are two thymine in the biology. One is THIAMINE, which is vitamin B1. but THYMINE is T, which is your here pyrimidine. Fine. Now, out of all these, the uracil will exclusively be found in the RNA, whereas thymine will be found in the DNA. Fine. Okay, let's see how a nucleotide is formed. So, first of all, you need to have one sugar. And here in sugar, for example, this is a ribose sugar. You will be having OH here, OH here. Let's label the carbons 1, 2, 3, 4, and the fifth one is outside the ring, CH2OH, right? Here it is. So on the first carbon, there will be formation of a glycosidic bond. There will be a formation of glycosidic bond with the nitrogenous base. So this bond is glycosidic bond. So the sugar is bonded with the nitrogenous base, which can be adenine, guanine, cytosine, thymine with the glycosidic bond. Whereas here, what will be there? Here again the water will be removed and phosphate will be attached. Okay. And here the bond is phosphoester bond. Which bond? Phosphoester bond. Since it is alcohol and this is acid, phosphoric acid, and whenever a bond is formed between the ester and the acid, the bond is the ester bond. Since because here phosphoric acid is used, so what kind of a bond it will be? It will be phosphoester bond. Fine. So this is one nucleotide. All right. Next. So we have to name these nucleotides. For example, if I'm using adenine, if I'm using adenine as a nitrogenous base, what will its nucleoside be known as? Adenosine. What its nucleotide will be known as? Adenylic acid, right? Because phosphoric acid is now added, so you call it as acid. If I'm using guanine, its nucleoside will be guanosine, and this will be guanylic acid. If I'm using cytosine, its nucleoside will be cytidine and nucleotide cytidalic acid. Thymine, thymidine, thymidalic acid. Uracil, uridine, and uridalic acid. And from these parts, a lot of questions can arise for exam. So be very particular in understanding these. What is a nucleoside? It's sugar plus nitrogenous base. What is a nucleotide? Sugar plus nitrogenous base plus phosphate. And whenever phosphate or phosphoric acid comes, naturally the name will be according to the acid, right? All right, you can please write this down in your copy. All right. Next, we are going to talk about DNA. What is DNA? DNA is deoxyribonucleic acid. Deoxyribonucleic acid. The sugar used is obviously deoxyribose. The DNA structure was given by Watson and Crick. Who gave the structure? Watson and Crick. So we follow the Watson and Crick model of bDNA. What type of DNA? bDNA. Because there are different types of DNA, A, B, C, Z. These type of DNAs are there. So if I talk about this Watson and Crick's model, according to this model, the DNA is a double helical structure like this. DNA is a double helical structure. It's a double helical structure like this. It's like there are two alpha helix. So that means it's a secondary structure with respect to the protein structure. These are two helix and they both run around one single central axis. So the first thing, it's a double helical, right? Second, anti-parallel. That means both the strands, they are opposite to each other. If the green one is 5 prime to 3 prime direction, then the orange one will be from 3 prime to 5 prime direction, right? So there are, like we say, these are forming, a double helical structure, and it's around a central axis. So here it's rotating for 360 degree. So we say it's one turn. This one turn is of 360 degree. In one turn, there are 10 base pairs, and the length of one turn, right? If I'm talking about one turn, that means 360 degree. That means 360-degree turn it is taking, like this. All right. Then if I talk about one turn, what will be the length? It's 3.4 nanometer or 34 Armstrong. If I'm saying there are 10 base pair, for example, I say there is guanine and then there is cytosine. So guanine will always be present in front of cytosine, and adenine will always be present in front of thymine in DNA, right? Both are present like in this manner. So here will be 1 2 3 4 5 6 7 8 9 10, 1 through 3 4 5 6 7 8 9 10. So here comes the 10 base pair. So that means if I say the distance between two base pair, what it will be? It will be around 3.4 around upon 10. It is 0.34 nanometer. So the distance between base pair will be, yes, it will be 0.34 nanometer or 3.4 Armstrong. If I say the complete turn is of 360 degree and we have 10 base pair, so every base pair will take a turn of how much degree? Then it will be around 36 degree. If I say the complete turn is of 360 degree, right? So one turn will be of, yes, one turn or ascent will be of how much degree? It will be 360 by 10, that is 36 degree. Okay. So each base pair moves at 36, 36, 36, and the complete turn of 360 degree. Alright. As I've told you, guanine will always pair with the cytosine and adenine will always pair with the thymine. In between these two are present hydrogen bond. If I talk about guanine and cytosine, there is triple hydrogen bonding. If I talk about adenine to thymine, that's a double hydrogen bonding. So that comes the next part, which says that guanine pairs with cytosine with three hydrogen bonds, and adenine pairs with thymine with two hydrogen bonds. So the hydrogen bonds provided the stability. So if somehow we increases the temperature, the hydrogen bond between these two strands will break and the DNA will denature. Next point. What is the width of this DNA from here to here? It's around 20 Armstrong. So what's the width? Width is 20 Armstrong. All right. So this was the helical structure of DNA. Let's talk about the molecular structure. So let's draw the molecular structure. So here we'll come to know what is a 5 prime and 3 prime. For example, if this is one strand, and here in one strand we have, since it is DNA, we'll be having deoxyribose sugar. So let's draw the sugar, but I will not be drawing everything, only the specifications will be there. So here we have H, we have OH, here we have CH2OH, fine, and here we have one nitrogenous base attached, fine. So here we have H, so we are going to remove the H because we are going to add phosphate here. This is one nucleotide. This is one nucleotide in a DNA. So how all these are linked and how it is forming the structure? If I talk about this structure of DNA, as you can see, the nitrogenous bases are present inwards, right? I have written G here, but actually the G is quite here and A is here. So we always say this total line which we have drawn, this is the backbone of the DNA and it is made up of sugar phosphate. What it is made up of? Sugar phosphate. So we say backbone is of sugar phosphate. Whereas these nitrogenous bases, they are hydrophobic. They are feared of water, so they will be present inwards. And nitrogenous bases, they are directed inward. Why? Because they are hydrophobic. All right. So now what we are drawing is this sugar and phosphate here, which we have drawn, they're going to form backbone, right? Similarly, we have to draw the another nucleotide here. Let's draw the another nucleotide. Okay. So here we have used adenine. Down here we'll be using guanine. All right. So again here CH2O. Then we have phosphate OH H. And here on the third carbon we have OH. Fine. This is what it is. So here we have first, second, third, fourth and fifth carbon. It's very important to label the carbon. You will get to know what's happening. Now, when we have to make the sugar phosphate backbone in DNA, what we have to do is we have to remove water. So, here we will be removing water. So, when we remove water, now just listen to me very carefully. Where are we removing water? We are removing water from third carbon's OH and phosphates OH, right? Third carbon's OH and phosphates OH. As a result, another another the another ester bond will be formed, but here you will call it as phosphodiester. Why? Because this is the second ester bond formed, because first phosphoester bond was formed here, right? When we have discussed the structure of nucleotide, many of you must have said phosphodiester. No, it's a phosphoester bond which is formed in a nucleotide or to form a nucleotide in between the sugar and phosphate. But when you have to make the sugar and phosphate backbone, or when you are making a bond between two nucleotides, so one sugar is giving its OH of the third carbon, another nucleotide is giving its phosphates OH, and this is how phosphodiester bond is formed. So one backbone has been formed, one helical backbone has been formed. Here in the fifth carbon, the OH, or you can say the phosphate, is not forming any phosphodiester bond, so this end will be 5' end. Whereas here, the third OH is not forming any phosphodiester bond. So this is the 3' end. And we always read from 5' to 3'. Do you get this point? You just have to see the fifth and the third carbon because they are forming phosphodiester bond. If the fifth carbon's phosphate is free, it will be 5' end. If the third's OH is free, it is 3' end, right? Let's make the anti-parallel strand just here. The anti-parallel will be drawn in the opposite direction. Obviously, we are drawing two deoxyribose. All right. So here, the third OH is free. So this is the 3' end. Okay. Now here, if we see, this is the first carbon, first, second, third, fourth, and this is the fifth one. The fifth one is having phosphate here. The phosphate is having OH, right? And here the first is having thymine because you know adenine will always be present in front of the thymine. And in this one, you will be having cytosine. And this third one will be having OH. The first one will be having the free phosphate. So that's why this end will be the 5' end. And we always read from 5' to 3'. So here we want to produce a phosphodiester bond. Let's make it. We just have to remove water. So it's very simple to make a phosphodiester bond. So here the another phosphodiester bond is formed. So this is how you can see the in the nucleotides, the nitrogenous bases are present inwards, whereas this backbone is produced by sugar phosphate. And now in between these bases, the hydrogen bonds are present. In between adenine and thymine, double hydrogen bonding. In between guanine and cytosine, triple hydrogen bonding is present. So that's the Watson and Crick structure of your nucleic acid, which is DNA. All right, moving further, let's talk about the Chargaff's rule. So Chargaff was a scientist who gave the rules, for DNA. Fine, rules for DNA. So what was a Chargaff's rule? First of all, the amount of adenine will be equal to thymine and amount of guanine will be equal to cytosine. This was the first rule. Second rule was amount of purine will be equal to pyrimidine, and then amount of sugar will be equal to phosphate. All right. So these were certain rules which were given by a scientist, and actually they only applied to DNA. And the last rule was the ratio. The ratio of nucleotide was fixed for a species, right? So what they said was adenine plus thymine to guanine plus cytosine ratio is fixed for a species. Is fixed for a species. That means if if we are human, if we are human, we have a fixed ratio. We have a fixed ratio means X of our all the nitrogenous bases. If there is bacteria, all the bacteria of that particular species will all be having the same that particular ratio. But a bacteria's and humans ratio will quite be different, right? All right. So this was about the DNA. Let's talk about another nucleic acid. That's RNA. What is RNA? Ribonucleic acid. Ribonucleic acid is formed of ribose sugar, as you know, and here there is no thymine. There are three types of RNA. One is mRNA, another is rRNA, and then the third one, tRNA. What's the difference? M means messenger, R means ribosomal, and T means transfer RNA. So all these are essential for the protein synthesis. For example, from DNA, the RNA is formed and you call this process as transcription. So mRNA contains message for protein synthesis. So it actually knows what type of protein will be formed. rRNA or ribosomal, it forms ribosome, which is a site of protein synthesis, which is the site of protein synthesis. tRNA, what is its function then? It's a transfer RNA. It will carry the amino acid from the cytoplasm and it will give to these two. And hence all the amino acids will join and form a protein. You will study this in the 12th class chapter molecular basis of inheritance, and that will be pretty clear to you. You just need to know what type of RNA are there. tRNA, it collects amino acid and transfer to other RNAs. Fine? Okay. So that's about the nucleic acid. Let's solve some questions. For nucleic acid, the building block is a nucleotide, nucleoside, ribose, nitrogenous base. One nucleic acid is a heteropolymer of nucleotides. So answer will be 1. Next. The bond between the phosphate and hydroxyl group of sugar in nucleic acid is a peptide, ester, hydrogen, and glycosidic. So it's phosphate, that means it's acid, and this is OH. Right? This is. So obviously the bond will be ester. When a bond is formed between alcohol and acid, directly if you don't know anything about it, you know the basic chemistry, you can crack this answer. So the answer is ester bond. Next, identify the structure and find the correct statement. It's a nucleoside and its name is adenalic acid. It's a nucleotide, the name is adenosine. It's a nucleotide and its name is adenalic acid. It's a nitrogenous base adenine. So here you have a ribose sugar, you have nitrogenous base, you have phosphate. So all three components are there. So definitely it's a nucleotide. So it's not a nucleoside, it's a nucleotide. And the name is not adenosine, the name is adenilic acid. So this is also incorrect. It's just not nitrogenous base, it's a nucleotide. So answer is 3. Next. Dynamic state of body constituents concept of metabolism. Right before the enzyme, a lot of pages are there which actually students, usually ignore them. So usually students ignore all these pages, but they actually contains a lot of information from where the question can arrive in your examination. So here the first point which we are going to discuss is biomolecules are present in certain concentrations, expressed as moles or cell or moles per liter, etc. One of the greatest discovery ever made was the observation that all these biomolecules have a turnover. This means that they're constantly being changed into some other biomolecules and also made from some other biomolecules. So what it says that dynamic state of body constituent concept of metabolism. We have discussed what is a metabolism. It's a sum total of all the chemical reaction. And we say our metabolism is dynamic. It's moving from one place to another or from one thing to another. So if we say we have dynamic equilibrium, our biomolecules are moving from one form to another. S1 has formed S2. Now S2 has converted into S3 and S3 into S4. So this is what is known as a turnover. That means the biomolecules are turning into one form or the another. They have been changed from one biomolecule to another. Fine. So if S1 is forming S2, later on S2 will form S3 and ultimately one end product will be formed. This is what this paragraph says. And it says biomolecules have a fixed concentration. For example, the concentration of glucose in blood is fixed. The concentration of everything in your body is fixed, right? Next. Metabolic basis for living, metabolic pathway. As we have discussed, what is a metabolism? Sum total of all the metabolic reaction. Now, in these, the metabolic pathway or reactions can be of two types. One are the catabolic reaction and another are the anabolic reaction. What's the difference? Catabolic means the destructive pathway. Here, the things are destroyed or broken down. Here, it's the constructive pathway. That means you are forming something. For example, I say in our liver, acetic acid is converted into cholesterol. So that means you're forming something, right? That's you're forming something, you're constructing something, that's anabolic. If I say the catabolic reaction where the glucose is broken down into pyruvic acid, that's your what? That's your destructive pathway, like glycolysis. So both the types of reactions are performed in our body, the one which destroy things and then which are producing things, because you need cholesterol for a lot of function. We have discussed it today. Also we need to break down glucose because we need energy. So two type of metabolic pathways are there, the catabolic as well as anabolic. All right, next. The living state is a non-equilibrium steady state to be able to perform work. So there is a sentence written in this paragraph around this entire this topic revolves. So we say that everything in our body is in a fixed concentration, and that is what steady state is. Steady state means everything is in a fixed concentration. For example, glucose. The concentration of glucose in our body is 4.5 to 5 millimolar in blood. It's fixed. If it moves, it gets, if it gets lower down or it gets, it increases in blood, it can lead to some metabolic disorders like diabetes. Fine. We say living state is a non-equilibrium steady state. Steady means everything is in a fixed concentration. Why we say non-equilibrium? Whenever anything is in equilibrium, this is how you write in chemistry. A is getting converted into B, B get backs to A. This is equilibrium. This is what equilibrium is. Our living state cannot afford the equilibrium state. Why? For example, A is sucrose, right? And sucrose is made up of glucose and fructose. I ate sucrose. I ate sucrose. If my body was in equilibrium, it must have formed glucose and fructose, and later on that will reunite to form sucrose. So in my body, is this going to give any benefits? No. I want glucose, so that glucose get broken down. What if the glucose broken down into pyruvic acid and pyruvic acid get back to glucose? You will not get energy, right? So that's why we say in equilibrium no work is done, but we want our work to be done. So that's why a living state is a non-equilibrium steady state to be able to perform work. If we say anything is in equilibrium, no work is done, but here we want some work to be done, so that energy should be produced, right? Okay, moving further to the next are enzyme. What are enzyme? Enzymes are made up of proteins, and some enzymes are also made up of RNA, right? So enzymes are mostly made up of protein. Mostly all enzymes are made up of protein, but some are RNA. These RNA, with time, with evolution, they have acquired some properties by which they can catalyze something, right? So RNA can also catalyze the chemical reactions. So these are ribozymes, ribozymes, ribonucleus B. These are the example. Fine. Like your telomerase, this is also a type of RNAs. So your enzyme, they are mostly made up of protein, and some are made up of RNA. Now, which protein structure does your enzyme exhibit? The tertiary structure, the woolen ball structure. When it rotates or move around, or when it is wrapping up in a tertiary structure, here comes the formation of a pit or a crevice or a pocket. What do you call this pocket as? You call it as the active site. So whenever it is the protein or peptide chain is getting converted into, what is a peptide chain? The chain of a lot of amino acid. When it is getting converted into the protein, it forms a pocket like structure known as active site. To this active site, substrate binds. What is a substrate? For example, sucrose has to get converted into glucose and fructose, and the enzyme uses sucrase. So, in that enzyme sucrase active site, the substrate sucrose will bind. Okay, so sucrose is a substrate. The product is glucose and fructose, and the enzyme uses sucrase. Fine. So, now these enzymes, you also call them as biocatalysts. Why you call them as biocatalysts? Because they are enhancing the rate of reaction. Enhancing the rate of reaction. You also have heard of catalysts that you use in chemistry. How they're different from them? First of all, they are in living organism, and those they are not in the living organism. Where are they present? They are in the normal chemistry lab or in that conditions. They're usually metals, and it's usually organic compound. That's a protein. Fine. Also, we say these enzyme, right? They get denatured at high temperature. Enzyme because they are proteins. Enzymes denature, that means it get broken down at high temperature and lost its tertiary structure. Now, what happened if it lost its tertiary structure? Then its biological function will be disrupted. But there are certain enzymes that are exceptions, except thermophilic bacteria. These thermo means temperature. These bacteria can survive at high temperature around 94 degrees Celsius. They are present in hot water springs. You know, you have seen usually near the volcanoes or in the hilly areas, there is a water pond like, and from there, heat is coming out. These are hot water vents, and there the bacteria, they have evolved themselves so that they can actually survive there. They can survive there in that high temperature. Fine. Okay, moving to next is a chemical reaction. So there are two type of chemical reaction. One are organic, which occur in living organism. Another are inorganic, which you study in the chemistry lab. In our body, I am talking about one chemical reaction, CO2 plus water reacts to form carbonic acid. They reacts to form carbonic acid. Fine. Now this carbonic acid, this this reaction, if it occurs without the presence of enzyme, how many molecules of this acids will be formed? Around 200 molecules per hour. But if I use carbonic anhydrase, so here without enzyme, if you perform this experiment, you will, or if without enzyme this reaction occurs in your body, you can only make 200 molecules of this acid in 1 hour. But if we use enzyme, which enzyme? Carbonic anhydrase. Carbonic anhydrase. Then we can produce lakhs of, yes, we can produce lakhs of these molecules in per second, right? So here is the thing. If you so if you use this enzyme carbonic anhydrase, around 6 lakh molecules are produced per second. 6 lakh is this much. Okay, molecules per second are produced. So what is the takeaway from this? We say this is a type of chemical reaction. So these biocatalysts or the enzyme, they are enhancing the rate of reaction. What they are doing is they are speeding up the reaction. If if there would be no enzyme in your body, I don't think so we will be able to survive, right? Okay. Moving further to the next, we have how do enzyme bring about such high rates of chemical conversions? Now it's a question, if they're increasing the rate of reaction, now how are they doing it? Let's talk about it. So if I'm saying that they are increasing the rate of reaction, right? Rate of reaction is actually what it is. Rate of reaction is amount of product formed per unit time. Amount of product formed per unit time. You also call it as velocity if the rate of reaction, or you can say the direction of the reaction, is specified. If you know the direction of a reaction, it is going in a particular way, you will call it as a velocity. So number of product forms per unit time is your rate of reaction. Okay, rate of reaction. Now how do enzyme how do enzymes do this conversion? So for that you need to understand the enzyme cycle first. For example, if this is the enzyme, this enzyme will bind to its particular substrate. Now after they have done binding, a transition state will be formed. So this is a transition state. Okay, in this transition state, what happens is it's a delicate state where a lot of bonds are broken or newly bond are formed. If I say this enzyme is sucrase and this is sucrose, so this is the substrate, this is the enzyme. So whenever enzyme substrate complex is formed, after that a transition state is formed where either the bonds are broken down or the bonds are formed. After this, if it is sucrase, it will break the bond and the products will be formed, right? So let's make the product. So these are the products. After that, the products will be released and the enzyme will be reused again. Enzyme will be reused again. So these are the products. Okay. So this cycle of enzyme will keep on happening in your body. For example, I have enzyme sucrase. It will first bind to one molecule. It will form ES complex where there it will pass through transition state. The bonds are broken, and then it will form enzyme product complex, and after that the product will be released and enzyme will be free to be used again. So we say enzymes are never used up in this cycle. You can reuse them as much as you want. All right, moving further to next, to the point that how do they do these conversions. So for that you need to understand a graph. If I have this graph, in this axis we have potential energy, and here we have the direction of reaction or progress of reaction. Okay, so the first graph I'm going to draw is when your substrate is getting converted into product and there is no enzyme, like this. All right, here is your substrate, here is your product formed, here is the enzyme substrate complex, the transition state. Okay, so to reach from substrate to transition state, we need a lot of energy. You can see this is the energy barrier because at this axis we have energy. So you need this much energy to reach at that stage. When enzyme comes, it decreases this energy barrier. Okay, so this is a reaction when enzyme is when enzyme is there. Okay, here enzyme apps. So you can compare the energy barriers of both these. Fine, you can compare the energy barriers. So what energy is this which enzyme is reducing? That is activation energy. What is activation energy? It is the energy that we need to start a chemical reaction. For example, there is one molecule, there is another, we want them to unite, right? They are just so much into a high energy that they're moving here and there. We need our hands energy to just push them or pull them together so that they will meet. So this is what activation energy is. What if enzyme reduces this barrier? Enzyme first it will bind to this substrate, and then to the another, and ultimately both have to unite. Fine. For example, both these molecules, if they want to meet, they require a lot of energy to come together. But what if I just pick up this substrate and pick up the another and let them unite? Then for those molecule it will be pretty easier. Fine. So this is what enzyme does. They reduces the barrier of activation energy. What is activation energy? Energy required to start a chemical reaction. Fine. So what does enzyme do? Enzyme reduces the activation energy barrier. Fine. So this one is activation energy. So if you can see here, this axis is energy. If you see this reaction, is it endothermic or exothermic? Substrate is at high energy level, product is at the lower. So when substrate is converting into product, a lot of energy is released. So this reaction is the exothermic reaction. But if the opposite occurs, in this what is there? Substrate at higher level. So it's the exothermic. If opposite occurs, the substrate is at the lower level and product is at the higher level, the reaction will be endothermic. All right. I hope that's pretty clear to everyone. So this is how the enzyme work. This is how they decreases the activation energy barrier. All right. Moving further to the next. Next, we have factors that affect the enzyme activity. There are a lot of factors that affect the enzyme activity. The first one is temperature. The first one is temperature. How does the temperature affect the activation energy? If I draw a graph between the enzyme activity, how the enzyme activity is affected by the temperature. So if I talk about the temperature, the which can be a very important factor, how it is going to affect it. For example, I've told you the proteins are forming the enzyme, or enzymes are made up of proteins, the tertiary structure of proteins. What if there is very high temperature? The protein will get denatured, then there will be no proper functioning of a protein. So whenever there is high temperature, the proteins get denatured, so enzyme cannot work to the its up mark. If the enzymes or if the temperature given to the enzyme is so low, it will get inactivated, right? So what happens is, you put your food in the refrigerator, right? Why do we put our food in the refrigerator? Because we want all those bacteria's enzyme to get inactivated, because at lower temperature the enzymes are not working properly, so our food do not get stale. So here temperature plays a vital role. So at this point we have temperature, and here we have activity, enzyme activity. As you can see, at this point the activity is higher, so that means this temperature down here, this one, right, is the optimum temperature. Fine. So at optimum temperature, so optimum temperature is a particular temperature where the enzyme activity is the highest. Why? Give me one example, or explain me with this example. For example, when you get fever, your body temperature rises and your digestion process, it actually doesn't work, doesn't function properly. Fine. Why? Because enzymes are not working properly, because the body temperature is higher. So we say there is optimum temperature at which enzyme activity is higher. Second is pH. Second is pH. Now what about pH? Same graph for the pH. There is always one optimum pH at which the enzyme functions. Okay, so here it is, enzyme activity. So here it is showing the maximum activity, like your gastric enzymes, they work at the acidic pH, and whereas your intestinal enzyme, they work at the alkaline. Third is substrate concentration. Third is substrate. So here we have a graph of substrate concentration. Down there on this x-axis, we are drawing the amount of substrate, and here the velocity. Okay, so if I have six enzymes, and I'm adding substrate, when I added one substrate, the reaction starts. I added another, more reaction start. I added another, the reaction reaches half. So that's Vmax by 2. Fine. Then what is Vmax? The maximum velocity where all the reaction has done or completed. Fine. Then I added more, and more. And at what time? At one time, the graph will become stationary. What is that point? Where all your enzyme is saturated by the substrate. Fine. The graph is becoming stationary there. Why? Because at this point, all enzyme is saturated. It is filled with the substrate. All right. Yes. So the substrate concentration where half of the reaction is completed, you call it as Km, or Michaelis constant. So what is Michaelis constant? It is that substrate concentration where Vmax by 2 is achieved. That means half of the reaction is completed. That substrate concentration where Vmax by 2 is, right? I hope this is clear to everyone. So, temperature, pH, and substrate concentration. These are three factors that heavily affect the enzyme activity. But there is one very important factor apart from all these, which actually stops the enzyme activity. So it is one of a factor that heavily affects the enzyme activity, that are inhibitors. Now, what are inhibitors? Any substances that reduces the rate of reaction or velocity of a reaction or Vmax of a reaction, that's your inhibitor. Substance that reduces rate or V. So we have two type of inhibition. One is competitive inhibition. Another is non-competitive inhibition. Now what are these? So inhibitors work by two ways, guys, right? Inhibitors work by two ways. So we have to discuss this in detail. So here we have two graphs. Both are the same, as you can see. These are depicting the relationship between the substrate and the velocity or the rate of the reaction. So what happened in competitive inhibition? In this inhibition, there is a competition going on between substrate and the inhibitor. Why? Because the shape is same and it can easily bind to the active site, right? So for example, if this is the enzyme, this is the substrate, and this is the inhibitor. The shape is same and it can easily bind to the active site. But what if I increases the substrate concentration? It will push away the product and it will bind to the active site. As a result, your Vmax will be achieved. Your Vmax will be achieved, but only when you increase the substrate concentration. So as a result, you will find out that because we are increasing the concentration, only then you are getting the Vmax by 2. So the Km now that will be more. So in this type of competitive inhibition, the Vmax is constant, whereas the substrate concentration is increasing. So likewise, Km will increase. So the best example to explain the competitive inhibition is, there is one enzyme, succinic dehydrogenase. Its substrate is succinic acid or succinate, and its inhibitor is malonate. So when we increase the concentration of succinate, the succinate pushes away the malonate, which is the inhibitor, and the enzyme is free to bind with its substrate and product will be formed. But what's that condition? Condition is that you need to increase the concentration of succinate. Fine. Now, what is non-competitive inhibition? In non-competitive inhibition, the enzyme have one active site and one allosteric site. So the inhibitor will not bind to active site. There is no competition going on between substrate and inhibitor. Rather, inhibitor is very clever and it will bind to this site, and this site is the allosteric site. Now, what is the allosteric site? The site other than the active site, right? So here the substrate cannot do anything even if you increase the concentration of substrate, because it is bound to that allosteric site. After it bind to this allosteric site, the shape of the enzyme's active site, it changes, and as a result, no more substrate can bind, right? So whatever you do, how much concentration you increase, ultimately your Vmax will not be achieved. So this is what happened in non-competitive. So in non-competitive inhibition, the Vmax decreases, Km constant. Fine. You need to remember all these things. These are very important. From these, these are like gems for solving the questions. What is the example? Inhibition of your cytochrome oxidase with the inhibitor cyanide. So in case of cyanide poisoning, you must have heard of cyanide, it's a poison. The enzymes of mitochondrial electron transport chain cytochrome oxidase gets inhibited because the inhibitor, this is inhibitor, it binds to the allosteric site and it will not allow the substrate, and hence no product is formed, no Vmax of the reaction will be released, everything is done, finished. All right, now let's talk about the classification of enzyme. So we have 6 classes of enzyme, as you can see here, and you have to learn these in the same sequence that's given. Oxidoreductase is that class where the oxidation or reduction reaction occurs to the substrate. Example, alcohol dehydrogenase. Alcohol dehydrogenase. So here what is happening? Oxidation or reduction, gain or the loss of the electron. In the transferases, there is a transfer of group. For example, A is attached to B, it reacted with C. Now A will bind to C and B will be released. So here we have transferred the group. What are we doing? We are transferring the group. So here transferases, we have one example, that is transaminase. Transaminase. Here which group is transferred? Amine group of amino acid is transferred. Hydrolases are those, for example, I give you example, sucrase. Sucrase on addition of water will break the glycosidic bond of sucrose, and hence glucose and fructose will be released. You know how the sucrose was formed? Sucrose was formed when there is a reaction between dehydration reaction between glucose and fructose. I told you in biomolecules all you have to do is you have to release water. Fine. So here sucrase does the same thing. When you add, you flip the reaction or you're getting it backward, you add the water, bond will be broken. Lyses also break the bond, but without water, without the use of water. Hence, you will see double bonds will be formed. Double bonds are formed in the product. Then we have isomerases. Here, one form of isomer is converted into another. In ligases, ligase means, here we have G. G means glue. It is, joining some things like DNA ligase. It join things. Okay. So these are the 6 classes. So I have told you to learn the sequence in the same manner. Why? There is a reason, because you need to know the nomenclature, right? So how do you name the enzyme? So there is a digit formula 1, 2, 3, 4. First digit tells the class of the enzyme. Second tells its subclass. Third tells its subclass. And fourth tell about the individual enzyme. But we have not done the subclass or subclass. So if, for example, in exam the question comes 2, 3, 4, 5, tell me, this is the nomenclature of enzyme, tell me for which class does it belongs to, right? So on number 2, we say on number 2 there is a class. Let's see on second number which class do we have? We have transferases. So that means this enzyme, it belongs to transferases. All right. If that is 1, 2, 3, 4, the first class is oxidoreductases. This belongs to oxidoreductases. So this is how the enzymes are nomenclatured. Next, the very important and the last topic of this chapter is cofactors. Now, what are cofactors? First of all, for that you need to understand what is holoenzyme. Holo means complete. It's a complete enzyme. Complete enzyme is made up of 2 parts. One is apoenzyme, which is the protein part of the enzyme, and another, the non-protein part, which you call it as cofactors. What do you call it as? Cofactors. So cofactor is a non-protein part. These non-protein parts, these are of 3 types. How many types? 3 types. One is a prosthetic group, Wherever the word prosthetic group comes, that means it's completely and tightly bonded. Second is coenzymes. And third is metalines. All right. If the enzyme lose its cofactor, the enzyme will not be able to perform its work. Prosthetic group are the one which are tightly bonded. Metal one are also tightly bonded. Why? Because they show coordinate bonds. Whereas the coenzyme, they are loosely bonded because they only forms bond whenever they need to catalyze a reaction. Otherwise, they will not form any bond. The prosthetic group, which is tightly bonded, we have one example: enzyme catalase peroxidase. These catalase and peroxidase enzyme, they have a prosthetic group named as heme. If the heme is removed from them, they will not be able to perform any function. Coenzyme, in coenzyme, we have the example of vitamins, like vitamins B. The vitamin B3, it contains FMN and FAD, right? And these are what? These are nucleotides. Without these, a lot of enzyme would not work. And their association with the enzyme is transient. They will only function when there is a need. Metal ions, we have like zinc. Zinc is a cofactor for carboxypeptidase. Similarly, magnesium. Magnesium is for hexokinase, right? So these were cofactors. We have three type of cofactor: prosthetic group, coenzyme, and metal line. Out of these, coenzyme are loosely bonded. All right, moving further to next. Next, we have questions. Which of the following factors affect the Vmax of reaction? Inhibitor, pH, temperature, all of these. So which of the following effects? Yes, inhibitor does it, pH does it, and temperature as well. I told you high temperature leads to denaturation, low temperature leads to inactivation. So answer is 4. Next. Enzyme catalyzing interconversion of optical, geometric, and positional isomers are the one which changes one isomer into another. Isomers are a different type. We have done it in the chemistry, geometric, optical, and all. So that is straight away question answer number 1. Hydrolases, they break the bond on addition of water. Ligases joins the bond, and oxidoreductases helps in oxidation or reduction of substrate. Next. Zinc is a cofactor for very, very simple carboxypeptidase. Answer 2. Hexokinase, we have magnesium. Calmodulin binds to calcium ions, and ferritin is for iron, right? So calmodulin and ferritin are the proteins to which your calcium binds to calmodulin in your body, in the blood, and ferritin is in the liver, and to this iron binds. All right. So that's it. Thank you so much. Please read the NCRT very carefully, and watch all this chapter. We have done a lot of things here, right, for you. And we have, like, I think I have given my 100% so that you can get a quick revision. And we have worked so hard to make this biomolecules a big hit to you, or big hit for you. That depends, it's hit to you or it's for you. All right, so please, you also work hard along with us. All I need is my students to work more harder than I do. Because it's your aim, it's your dream. All I can do is I can show you the path you have to go, or where you have to go, or I can tell you this is the way you can do it, or I can explain you things. But ultimately, it's your aim. You have to revise, you have to learn, you have to watch the lecture religiously, and you have to stick to what you have you have ever dreamt of, like becoming a doctor or anything. But be true to that. I'll meet you in the next class. Till then, bye bye. Thank you so much.